Yes, sir. Hello. Yes, sir. Please, please. Rada ma'am.
यस सर, मेरी आवाज आ रही है सर आपको?
Yes, ma'am.
Yes,
Clear.
ma'am.
The biggest innovations of the 21st century are at the intersection of biology and technology. A new era is beginning. Very warm good morning to all of you. According to Indian tradition, every work begins with the
Mic mute.
Am I audible?
Yes, ma'am. Yes, ma'am.
It is requested to all, please mute yourself. The biggest innovation of the 21st century are at the intersection of biology and technology. A new era is beginning. Very warm good morning to all of you. According to Indian tradition, every work starts with the worship of Ma Saraswati, the goddess of knowledge. So let's start this event with the prayer of Ma Saraswati. हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ, हे शारदे माँ, तू स्वर की देवी, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे, तू स्वर की देवी, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे, हम हैं अकेले, हम हैं अधूरे, तेरी शरण में हमें प्यार दे माँ, हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ, अज्ञानता से हमें तार दे माँ, अज्ञानता से हमें तार दे माँ. I, Dr. Prerna Dubey, extend a warm welcome to all respected guests, respected dignitaries, and participants virtually present here. First International Conference on Challenges and Prospects of Biotechnology, Environment and Agriculture Researches, CPBEAR 2022, is being organized by Department of Biosciences, Christian Eminent College Indore, in joint collaboration with the Research Foundation of India. Christian Eminent College has achieved a remarkable place in the field of education in the journey of 25 years. At present, 1 diploma program, 5 PG level and 11 UG level courses are being offered by the college. And the Research Foundation of India is a registered organization which continuously conducts seminars, conferences, and workshops across the globe. Under MOU, the Christian Eminent College has conducted various activities in association with RFI. Today's conference is sponsored by JHERF, World Virtual Conference Forum, World Federation of Science and Technology, International Research Guide Federation, Academic Research Guide Association, and Edu Academic INC. To inaugurate this conference, today we have with us respected Dr. S.K. Mishra Sir, Principal Scientist and Head in Charge Genetics and Genomics Laboratory, ICAR, Central Avian Research Institute, Regional Center, Baramunda, Bhubaneswar. Respected Dr. Supriya Kumar Vellan, International Speaker, Communication and Leadership Coach, Soft Skill Trainer, Global Motivational. Respected Dr. Sudhakar Umlesar, Head and Associate Professor, Mechanical Engineering Department, Sardar Patel College of Engineering, Andheri, Mumbai. Respected Dr. Pratik Rajan, sir, Scientist, Professor, Global Educator, Counselor, Published Author, International Speaker, and President of Youth Development, IIU. Respected Dr. S. Dragon Scopia, educator, speaker, lecturer, inventor, scientist, researcher, social worker, author, phytotherapeutics, phytonutritionist, agroecologist, natural health coach, Scopia, Macedonia, and respected Dr. Rania Lampo, global educator, STEM instructor, and an ICT teacher trainer in Greece as eminent guests and speakers. Hearty welcome to all of you. I also welcome to entire management and administrative authority of Christian Eminent College Indore and Research Foundation of India. The Research Foundation of India is a registered organization working for the social upliftment and higher education quality and Indian research promotions. RFI works for the integration of intellectual ideas across the globe. RFI members are faculty, researchers, graduate students, and other distinguished professionals with rich and diverse expertise in education research. It has signed MOU with the top universities from India. Under MOU, the Christian Eminent College has conducted various activities in association with RFI. Chairman and CEO of RFI, Dr. Saurabh Jain sir, is among us. Saurabh Jain sir, are you present here? Jain sir? Okay. I think some technical issues with Saurabh Jain sir. Now,
मैम
moving
सब
ahead.
ज्वाइन कर रहे हैं थोड़ी
Yeah.
देर में हमें.
Okay, okay. The success of any educational institution is determined by the impact it creates in young minds. These are the minds that will one day grow to become the pioneers of this generation. A small sapling of inception in 1996, the Christian Eminent College grew exponentially into a large multi-branch tree within a few years, achieving several milestones. Christian Eminent College is embarking onto new avenues of teaching learning experience which make it very different, creative, and fruitful. The UGC has granted autonomous status to the college. In addition to that, the college has also been awarded cycles. Christian Eminent College emphasizes on skill building and overall development of learners. The college students have excelled in the academic as well as the sports and cultural activities. They have given outstanding performances at state, national, and international levels. Christian Eminent College is known for its excellence and talented faculties who have conferred on with a number of national and international awards. Christian Eminent College also strive for the best. Now, moving ahead, I would like to request Professor Rajesh Shah sir, Honorable Vice Principal of Christian Eminent College, Indore, for address.
Thank you, Dr. Prerna Dubey, madam.
Welcome,
Thank
sir.
you. A very warm good morning to one and all present here. On behalf of NAEC A-grade accredited Autonomous Christian Eminent College, Indore, I, Professor Rajesh Shah, Vice Principal, heartily welcome all our distinguished guests in inaugural session of this three-day international conference. This international conference on challenges and prospects of biotechnology, environment, and agricultural researches is jointly organized by Department of Biosciences, Christian Eminent College, Indore, and Research Foundation of India. I heartily welcome and thanks all our renowned international and national guest speakers, Dr. S.C. Dragon Jovano from Macedonia, Rania Lempo from Greece, Dr. S.K. Mishra from Bhubaneswar, Dr. Supriya Kumar Velian from Chennai, Dr. Sudhakar Umble from Mumbai, and Dr. Pratik Rajan Mungekar. Thanks to all eminent guests and speakers. I would like to thank and congratulate Dr. Saurabh Jain sir, young and energetic chairman and CEO of Research Foundation of India. I also express my sincere gratitude and heartily welcome to founder member and CMD of RFI, Professor Dr. Ashok Kumar Gupta sir. My heartiest welcome to Dr. Sanjay Maheshwari sir, Vice President, MP State Student Wing, and my very special thanks to my beloved friend, always smiling, Dr. Ajay Jain sir, National Coordinator RFI India, for his constant support and coordination. I also thanks and welcome all participants of this conference. My special gratitude to management of Christian Eminent College, our chairman, Honorable Mr. Samir Mir sir, Director Professor H.N. Gupta sir, Director Admin Professor Y.S. Chauhan sir, and Principal Dr. Rajesh Vyas sir for their constant motivation and support. Last but not the least, I would like to congratulate Dr. Shivam Pratap sir, HOD Biosciences Department, Christian Eminent College, his department members, and all members of organizing committee. I am very sure that this conference will be a grand success. My best wishes to all. Thank you all again. Thank you very much.
Thank you
Over to
very
Pratik.
much, sir. I want to say something about Shah sir. Shah sir is having more than 27 years of academic and teaching experience. He is the life member of the Indian Society for Technical Education and National Geographic Society, member of Board of Studies, Computer Engineering and Computer Science, Devi Ahilya University, Indore, member of Advisory Board of Diversifications, Diversifications Digital India, Cashless Movement, Cleanliness Drives, and Voter Awareness Programs. Thank you very much, sir, once again.
Thank you.
Now, I would like to request member and CMD of Research Foundation of India, respected Dr. Professor Ashok Kumar Gupta, sir, for address. Dr. Ashok Kumar Gupta sir is having a rich experience in the industrial field and educational field. He had published more than 50 research papers in national and international journals. Professor Dr. Ashok Kumar visited USA and renowned universities. He is one of the founder member of Research Foundation of India. Over to Dr. Professor Ashok Kumar Gupta sir. Please say a few words.
Thank you, ma'am.
Over to Dr. Gupta.
Hello, everyone. This is Professor Dr. Ashok Kumar Gupta, CMD RFI. I feel honored to be invited to this inaugural session of International Conference on Challenges and Prospects of Biotechnology, Environment, and Agriculture Researches. The conference aims to provide the most complete and reliable source of information on the recent advancements in the field of challenges and prospects of biotechnology, environment, and agriculture researches. The emphasis will be given on publishing quality articles, making them available to the researchers and the young scholars worldwide without any restriction. This webinar on challenges and prospects of biotechnology, environment, and agriculture researches covers a broad range of topics in this field. The National Education Policy 2020 has given a clarion call for holistic value-based education to prepare our youth and develop our nation. Education is fundamental for achieving full human potential, developing an equitable and just society, and promoting national development. Now, it's my privilege and honor to welcome all the dignitaries present over here. I'm thankful to the entire team, RFI and Organizing Institute. I'm thankful to all who have given their input in this conference and all participants who made this conference a success. For more information, you may contact through our official mail info@researchfoundationofindia.com, and for such other events like this, you can visit our website www.researchfoundationofindia.com. Thanks for your patience. Thank you again. Over to you, ma'am. Thank you.
Thank you very much, sir, for your kind words. Now, I would like to invite Dr. Shivam Pratap, sir, Organizing Secretary of Conference Committee and Head Department of Biosciences, Christian Eminent College, Indore, to enlighten the purpose of International Conference on Challenges and Prospects of Biotechnology, Environment, and Agriculture Researches. Over to Shivam, sir.
Good morning, ma'am. Ma'am, am I audible?
Yes, sir, you are eligible.
A very warm good morning to all, to Honorable Professor Y.S. Chauhanji, Director Admin, Dr. Rajesh Vyasji, Principal, Professor Rajesh Saha, Vice Principal, Christian Eminent College, and Dr. Saurabh Jainji, Chairman and CEO, Research Foundation of India. Guest speaker, Dr. S.K. Misraji, Principal Scientist, Regional Central Avian Research Institute, Bhubaneswar, and distinguished national and international guest of speaker, along with Dr. Ajmat Alam Khan, Professor cum Chief Scientist and officers on special duty to Vice Chancellor, Sukast, Kashmir. I, Shivam Pratap, the Head Department of Biosciences, feel proud privilege to welcome you all in our international conference, Challenges and Prospects of Biotechnology, Environment and Agriculture Researches, CPVER 2022, jointly organized by Department of Biosciences, Christian Eminent College, Indore, and Research Foundation of India. Now, the question may be why we have decided to go with the topic challenges and prospects of biotechnology, environment, and agricultural researches. This time, each of you may have your own opinion, but we have selected this burning topic due to the following reasons. The prime objective of this conference is to provide a versatile platform for knowledge sharing among the eminent speakers, scientists, academicians, and delegates, especially to our youth of biotechnology, chemical sciences, agriculture, and PhD scholars from versatility in domains. Our goal is to build awareness in our youth about the current status and career prospects in biological sciences and allied fields. We hope that the presence and knowledge sharing of our invited national and international guests will definitely be a roadmap and great value addition to our youth. Due to COVID-19 pandemics, knowledge sharing has been restricted in different ways, which is rely on the online education right now. So the realistic experimental interaction are affected somehow, that such lacking of researches and interaction with eminent faculty may overcome with the help of this ongoing conference and symposiums. For wider prospect, we have included certain sub-themes for the betterment of our students and delegates, like advances in agricultural researches, emerging environmental challenges, and sustainable development, biofuels, microbial biotechnology, genomics and proteomics, and molecular markers as well. Various challenges still remain, like the employable biotechnology professionals, lack of experimental learning, lacking of fruitful researches, conceptual learning is going to deteriorate right now, limited job opportunities, especially in government sectors, green and certain environmental issue like greenhouse effect, destruction of ecosystem, problem in pharmaceutical industry like establishment of R&D sectors, etc. We are quite hopeful that such interaction in terms of 3 days of this international conference will provide the fruitful suggestions and answers will resolve above issues. I am so fortunate by having my PhD supervisor, Dr. S.K. Misraji, with us in our conference. who are my idol, and I have learned lot many things from them. And he supported me a lot during my PhD work. I will remain highly adapted for that throughout my life. We are looking forward to forthcoming invitee talks and technical sessions during entire 3-day international conference ahead. I express my sincere gratitude to all the delegates for successful and fruitful outcome. All the very best and have a good luck to all the participant delegates, eminent speakers. Thank you, ma'am. Thank you very much. And over to you, ma'am.
Thank you very much, sir, for throwing the light on the theme of the conference. Thank you very much once again. Now, we are greatly honored and feel privileged that respected Dr. S.K. Mishra, sir, Principal Scientist and Head in Charge Genetics and Genomics, Laboratory ICAR, Central Avian Research Institute, Regional Center, Baramunda, Bhubaneswar, has graced this occasion with his presence. Dr. Mishra is a doctorate in animal biotechnology from UNIVAS, Chennai. Dr. Mishra joined ICAR as a scientist in poultry science in 1991 and is now posted at ICAR DPRS General Station, Regional Station, Bhubaneswar, as a principal scientist involved in genetics and genomics. He has wide international exposure through visit of 7 countries, including USA, Europe, China, UAE, Korea. Dr. Mishra completed 10 projects from ICAR, DBT, DST, NASF, NHI, DPR, Hyderabad, and CARI, Izratnagar. Dr. Mishra published over 75 research papers in various national and international journals. He is the member of several international, national scientific committees, professional organizations, and other bodies. Sir has been the recipient of many honors, awards, fellowships like Junior and Senior Research Fellowship, Gold Medal in Animal Biotechnology 2000, Best Poster Presentation Award, Best Research Paper Award, Kalinga Ratna Samman 2016, PK Pani Award, Best Genetics Paper 2015, and many more. It's a matter of great pleasure and honor that such a distinguished and eminent personality is among us. Sir, we with immense pleasure welcome you wholeheartedly, and I don't take much time and requesting, sir, please share your knowledge with us on advances in genomics and biotechnology of dupes, the next important alternate poultry. Respected Dr. S.K. Mishra, sir.
Namaskar.
Sir.
Am I audible, Madam Dubeji?
Yes, sir. Yes, sir. You are audible.
Thank you very much. First of all, a warm welcome to all the participants here and respectable organizers. I think Christian Eminent College, RFI, and all of you, and whose name I'm not able to take here. All of you, sir, I'm thankful to you for giving me this opportunity to be along with you. And special thanks to my worthy doctoral student, Dr. Pratap, a wonderful candidate, and I believe he must be proving their worthy in his worthy manner. It's an opportunity to talk something on a relevant topic that I am associated with. If I'm correct, Dr. Pratap, how much time I have with me?
50 minutes.
Okay. So roughly if it is 11:25, I believe I can 12:05, 12:10, I think I can conclude.
Please continue.
So can I start the screen sharing?
Yeah. Yes, sir.
Screen is still visible. Just a minute. I'm just doing it.
Yeah.
Not sure it has come properly or not. Window. Yeah, I believe it has come now. Dr. Pratap, just confirm it is
Yes,
come now.
yes, sir. It is
Yeah,
visible now.
yeah. Coming 30-40 minutes, we are going to talk on this particular
Yeah,
aspect. And it's always a
right, sir.
pleasure that having understood the biotechnology and its theme, fitting to this the conference themes, we wanted to put on some challenges. What are the challenges facing one of the sectors that I'm associated with? Like already, Madam Dubey has already said that I'm associated with poultry, and we have received the trainings and we have done research on where the biotechnology can fit into this poultry genomics and the poultry world. Poultry industry is already well defined. Without the broiler chickens and eggs, there is no daily life. Even small to smallest to eldest and the family, they everybody knows that the family is missing a chicken, I mean, an egg omelette or a chicken item in the feast or any dawat, it loses relevance. So that much popularity the poultry has already got it. Without us, it is already there. So what else we are contributing to our knowledge and knowledge of biotechnology and genomics? So because I am associated with a sector that is dealing with the ducks, I'll take only 2 minutes introducing ducks, why I have chosen ducks to be a model species here. But in general, we'll be talking about the relevant principles of genomics, how that fits into the advancement of this knowledge. What is biotechnology? Until yesterday, until 2 to 3 decades before, when biotechnology was just happening, we were studying genetics in the same Mendelian fashion. Keeping Mendel in mind, we are trying to study quantitative genetics. Dr. Pratap has himself done a quantitative genetics study there, and he was also fortunate to have done some molecular techniques there. One of the wonderful students, he will also agree with me. We have to have a common meeting point between the quantitative tools and the modern genomics and the biotech knowledge that is upcoming every day. Every moment it is modifying and renewing. Where and how we are able to fit in with Indian context has to be seen. Let me spend a minute talking about ducks. Why we have chosen ducks? Because the poultry industry has been already established, I say is already significant. And the current shift is that from the backyard venture until 3-4 decades before, when I was a child, people used to catch a native chicken to serve a chicken dish. And all of you remember that we have to take the chicken with both hands because that was tough. Nowadays, just see the contrasting scenario. Any broiler item, chicken 365 or chicken tandoori or chicken tangri, you don't use the even hand also. You just use a toothpick. And in the starters, you see the maximum rush in a particular feast or dawat. Maximum rush will be there in the starter section where chicken will be there. Because it's so tasty. Reason what changes have happened here? The juiciness, tenderness, the growth potential has come through lips and bones. What was a tough chicken giving hardly 1 kilo, 1.5 kilo in a 3 months, 4 months period or 6 months or 1 year, let's say. It is now available within 4 to 5 weeks. People often confuse that people are adding some enzymes and doing something, some hormones to make the chicken softer, tender. I always try to overcome this canvassing. Because these are baseless. The modern chicken has been built in a manner. It has to grow. It has to rapidly proliferate itself so that it gives us quality protein and qualitative taste also. So with this background, chickens have already established itself. I am now talking of ducks. Why ducks? Because ducks are this next alternate poultry next to chickens. And world over, ducks are adored everywhere. You talk of China, whole of Southeast, your Vietnam, your Indonesia, Philippines, Korea, Japan, your Burma, everywhere they are mostly duck lovers. And duck is a domesticated poultry. It is a waterfowl and also equally productive like chickens. Whole of Europe, they have a different taste bud. They love different delicacies. Just chicken and just mutton, they will not like to take. You must have heard an item called foie gras. That's made out of duck liver. A duck that is having 1.5 kilo or 2 kilo can give you a liver alone of 400 to 500 grams. Sounds wonderful, no? But that is how the Europeans believe to take it. French love it. The Spanish people love it. The British love it. And in whole of USA, they are also duck meat lovers. I had been to USA a couple of times. I have seen how they make, they love chicken, but ducks for their meat potentials. So that is how it has become famous in USA. But and particularly in contrastingly in India and other Southeast countries, ducks are famous for eggs. So with these two potentials in mind, we all realize that ducks lay ducks are better versions of the domesticated poultry because they lay more eggs. Always, if you compare the best of the duck germplasm in the world, you'll find that ducks are laying at least 4-5 eggs higher than the chickens. Modern chicken means white leg chicken. How much they lay? 300 to 320 in a biological year, 365. Ducks lay more than that. 300 eggs I have written here, but it is actually more than. Ducks eggs are bigger in sizes. Roughly 70 grams, this size starts from 70 grams by the time it comes to market. Often you find eggs also weighing around 80 grams or above. 2 eggs, you consume duck eggs, and almost you are done with your meal or lunch or so. So with so much of attributes with the ducks has a red muscle fiber content. People say it's a potentiated meat, energy rich meat, antioxidant rich meat. One has to actually, we should do a lot of research to emphasize on that. But I believe the whole of China, they are biggest exporter of chicken, duck, and eggs, but they consume in-house, morning, afternoon, evening. All 3 times given a chance, they will like to consume ducks. Ducks, this meat, ducks in the salad, ducks in this roasted duck in the evening. Every time ducks. When I ask somebody in the Beijing, why do you love so much ducks? They say, you have to consume it to become like us. So Chinese people, they are known for their agility. A person in 70, they are able to use the staircase and climb 4-5 stairs. We require lifts. What is the secret of their agility? They say, sir, we are agile because we love this waterfall meat. Ducks and goose. They are packed with such vitamins, minerals, and above the antioxidants, which are very much essential to keep us agile and have a quality life. So that importance in mind, people started using ducks, and world over, ducks are also taken as a backyard poultry version because it doesn't require backyard ducks. They don't wait for you to feed them. They are able to survive on taking the fallen grains, all this dhan, gehu, all this that falls in the field. Ducks are able to graze there, and they eat on the snails, earthworm, insect, food waste. They rarely and hardly avoid for your feeding. So they are easy and they are hardy also. They are easily resistant, and they are interestingly they are resistant to most of the chicken diseases. So many attributes. Only you talk of Marex, it's not required. You talk of Newcastle disease, you talk of Gumboro disease, that's not there in the ducks. They're naturally resistant. And nature has endowed them with resistance for a purpose. All this, wastelands, all the water bodies, Nadi, Nala, Talab, Bandh, river, everywhere, which you are not able to utilize for any productivity. Many times people closer to sea, they must agree, some saline water seeps in there, it becomes brackish water, no more suitable for paddy and gehu. That's good for ducks. They can go inside, use that entire water body as a grazing ground and give you some productivity. What is productivity? Egg and meat they give you. Your bullocks will not go there, your sheep and goat will not go there, your chicken won't go there, but the ducks can go there. It's a beautiful bird, no? So with this background, let's talk why it has amused and aroused the interest of the whole world buying an important alternate poultry. And this is some small key statistics. I am not going to elaborate them, but just wanted to drive home a point. 1.14 trillion ducks are there in the world. 1.14 trillion. That means 1,014 billions. Okay. And Asia holds more than 89%, more than 90%. It supports the entire world's duck population. Where there? Obviously, China, Indonesia, India, Burma, your Philippines, and Vietnam, small country Vietnam, packed with ducks here and there. And for duck egg production, the native ducks, they normally hardly give eggs. 40, 50 eggs, they go bloody. You're having a flock of native ducks, but you don't have the egg production there. So relying on the native ducks, native chickens, you cannot make a carrier out of them. But you have a flock of native ducks, they will graze in the field, they won't demand for your food and still give you some eggs. Double or triple the capacity eggs they can give you. That is how whatever little attention has gone to ducks, we are not giving a lot of scientific attention. Whatever duck populations are available in India, most in the coastal states, they are all concentrated in the coastal states. Assam, West Bengal, Odisha, Tamil Nadu, Kerala, they love duck eggs, duck meat. And they give you a good production of roughly varying from 110 eggs to 170 eggs. So that much about ducks, and total duck population in India is 33 million, 30.5, that means 3.3 crores ducks are there. And production wise, they are able to give us this much potential, 180 eggs we are able to get in the farm ducks. And more research we do, that will all go towards one or two more slides, then we'll go to hardcore genomics. What are the different duck breeds available in India? Khaki Campbell, white Pekins, and native. Khaki Campbells are equivalent of your egg type. What is white leghorn to you? That is the white Turkey Campbell's to us. They're egg-laying machines, just a little bit lightweight. White Pekins are the broiler version of the ducks. Native ducks, the Chara, Chambeli might have heard, Nageshwari, Kuji, Norisa, small ducks, but able to give 75-80 grams of eggs. Small, just a small duck, 1.2 kilo, able to give a 75 gram eggs every day, 80 grams egg, bigger than your chicken eggs. Interesting, no? And these other ducks, Moti ducks, there is a Muscovy duck. It looks like a duck, but basically it's not a duck. It's a little bit different species. Genus wise also different, but I'll just come to that later. And these are other 3 duck breeds which are not there in India, internationally popular: Runner, Rowan, Aylesbury. They're not there. And these are the pictorials, self-explanatory. I'll just share this slide with Dr. Pratap, and he can distribute to you. So no need to exactly look at, but point to say that these ducks, many people have a misnomer that ducks means they require water. Actually, they don't need. Ducks are just like any other poultry. They can be reared in the same house where I'm sitting. All that you require is that just feed them and give them water for their consumption. They love playing in water. That's a different thing. We want to be a nature lover. We can give them water bodies. They breed well in the water, but doesn't mean that essentially they need water to survive. They can just survive in any deep litter. Backyard, if you leave, you give a water body, they will play there because they try to dip their head there. So that much about the introduction in ducks. And there is a native breed which does not require any feed almost. It is able to amass its own feed by grazing in the backyard. And these are your Muscovy ducks. They can go into 4-5 kilo. They can survive for 18-20 years. Given in a zoo situation, they can survive that much. Such beautiful species, how much justice we are doing and how much research we are doing. Little bit, let's talk about the duck genetics and cytogenetic map. All of you know that the chicken genome has already been sequenced. It was probably the first domesticated animal genome to be sequenced in year 2004. And nearly 16 to 18 laboratories they were engaged in sequencing the chicken. In those days, chicken means dideoxy sequencing. 500, 600 base pair read will come from one reaction. So at a time, 18-20 labs, they used to collaborate with 400, 500 people, all doing everyday sequencing part like that. How they used to do? Digest the entire chicken genome, make into different libraries, and they try to make small reads, and they try to align them. And likewise, they are trying to build the entire 1.4 billion, 1.5 billion, something native chicken chickens 1.4, 1.5 billion. That many base pairs, they used to align them manually. Nowadays, sequencing is different. De novo sequencing. Human genome can be sequenced within few hours. Totally, today morning you go, by afternoon you are done with your sequencing. They will give you a huge big cassettes of 10 or 12 DVDs, IDs DVDs, and they will say, this is all your genome sequence. How much you pay? Maybe you are paying around close to 2 to 3 lakhs rupees and getting your own genome sequences. So that much revolution it has come. But when it was done, why it was done? Because we wanted to study not only sequencing of ducks at par with chickens, but we want to align them and make meaning out of it. So they were concentrating on the cytogenetic map. Where you start the map? You start at the cytogenetic level. Within the chromosome, within a cell, there are chromosomes. Everybody knows that nucleus is the storehouse of chromosomes. If you make a metaphase spread and spread out all the chromosomes, how they look like, I'll show you in the next slides. So this started in the year 1966, where they realized that the diploid chromosome number in the ducks were 40 pairs. Chicken, how many? 39 pairs. I'm talking side by side chicken and duck, so it will be interesting to students also. back sequence data. Always use chicken data as the best and try to find out the differences between the chicken and ducks. And that here they are able to align sequence. If this is chicken, chromosome 1 of chicken, chromosome 1 of duck, how it looks like. Chromosome 2 of chicken, chromosome 2 of ducks. Likewise, they were able to assign and make comparisons. So they made that a sex average map spanning around 1353.3 centimorgans. They tried to find out, and this particular gentleman, Wang et al., that group, they published this map with coverages around male and female maps, something like 14.15 centimorgan and 13.87 centimorgan in the male and female perspective. Why this difference? Little bit difference, all between the G and W. G and W that was making the difference. So what else will be the difference? So this showed that cross-species homology between ducks and chicken sequences can be used as a background for integrating genetic and scientific. You don't know anything. You just given ATGC in that sequence. There is a huge millions and billions of base pairs. How do you integrate? How do you assign that this particular sequence belongs to chromosome 1, this much to chromosome 13? You need to have a base. So chicken was used as a base. So authors made use of 228 back clones and rest of that. They try to make back libraries. So then from the physical maps, cytogenetic map, people move to linkage maps. Linkage map, that means a linkage group. What is linkage group? The number of genes, number of markers, don't misread me, I'm going to talk to markers, come to markers later. The group of genes coming simultaneously, they are called to be linkers. I mean, it's supposed to be one chromosome, then only they can go together. En masse. 100 and 500 genes or sequences which are in linkage. Loci that is in linkage. They inherit en masse, go en masse. Nothing that is but a linkage, and that can be said they are coming from one chromosome. Maybe they are all in one microchromosome 16. They are coming en masse. So that is how linkage map was relevant. How did they build up? Using AFLP markers. What is AFLP marker? I'll tell you in the next slides. But used this and they made a marker that is linkage. They made 32 link groups used by 260 co-dominant markers. Again, what is co-dominant? And also I'll explain that. Co-dominant, that means both the version of the markers are available for the scientists to read. If one had to compare results generated from both these marker maps, that is, people have made microsatellite maps also in chickens. People made microsatellite maps in the ducks also. Somebody made AFLP markers, and this AFLP marker and microsatellite marker till the duck genome was sequenced in 2014. They were the basis for all the studies. And they made linkage groups. And Skinner et al., they made a map based on FISH, fluorescent in situ hybridization. You know that? Fluorescent tags you use to define, and you are able to have different colors. Some say painting probes or whatever. Your entire chromosome will be given different fluorescent tag markers, and you just make a big map in front of you, which will be linkage map essentially. And along with the concept, there is another thing you are hearing: copy number variations. This is another kind of markers. What is copy number variation? That also I'll tell you in the next slide. It's a higher version of the mini satellite. That means people must have heard the variable number of tandem repeats. That means core sequences being repeated number of times. Let us say ATGCA or TTGCA. TTGCA being repeated 5 times, 10 times, 100 times will make a repetition. One person has a 99 repeats, another person has 88 repeats. They look different by sizes, and we have, through our techniques, we can identify which particular animal has 88 and which particular animal has got 99 repeats. But if you make a very high, big repeats, let us say 10,000 base pair repeat. In one block, chromosome 1, 10,000 repeats, same chromosome 10,000 repeat is there in the other chromosome, third chromosome or the sixth chromosome. There is a copy number variation. Such a big repeat has been different and copied number of times, and that can be also used as a marker. So for the big mapping or the chromosome mapping, linkage, the CNVs have been often used. And interestingly, CNVs has been realized that chickens and most of the species, ducks, there may not be number of CNVs, may be very less as compared to other mammals. But CNV is very exclusively used in the mammals also, humans also. So summarizing the comparison of CNVs between chicken and ducks, extend the chicken and turkey. They made comparison between chicken and duck and chicken and turkey. And they said that avian genomes in general contain fewer CNVs than mammals, where genomes of evolutionarily distant species shared many regions of copy number variation. This is a typical comparative map of the chicken and duck chromosome. Okay, left side is the chicken chromosome GGA1, Gallus domestica, GGA, Gallus. Here, Anas platyringes, AP. AP means duck map. So chromosome 1 of ducks will be like, chicken will be looking like this. Chromosome 1 of ducks will be looking like this. What you see, almost physically same, little bit rearrangements. This portion might have gone here, this portion might have gone here. That means, where from this entire thing, if we believe in Darwin's theory, we all have evolved from a small moss, no? Sivli. In Oriya we call Sivli, in Hindi, I don't know what the word is. From that, we have all come up through different evolutionary routes. We might have been same millions and millions of years before. Primates and humans, they have evolved thousands and thousands of years before. Not thousands, again millions, all down from settle down to million. I don't know whether millions years before there are human beings or not, but then definitely some life organism was there from which all of us evolved. There is a big dispute because no one has seen millions of years. Technique says that carbon dating and all other techniques, they tell us millions of years before these people evolved like this. It's plausible also. How else animal primates are there, humans are there, and genetically, chromosome-wise, DNA sequence are almost same, but looking different. It's evolution. And the evolution comes this way. The same chicken looks like this, the ducks look like this. Little bit means rearrangements. And this, if you just different chromosomes you organize, you find different forms of rearrangements. Okay? GGF4 corresponds to APL10. That means fourth chromosomes component was identified as 10th chromosomes component in the ducks. Bulk of that, not all. This Skinner et al.'s theory. Rao et al., another group of an Indian American, they make a duck whole genome hybrid, radiation hybrid panel. What is radiation hybrid? It takes some time. I think Dr. Sivam can expand that later. So that means they try to fuse by making a radiation. You make embryonic fibroblasts irradiated at the dose of 6,000 rats with HPRT deficient cell line from the hamster cells that are fused with the duck sequences, and from that they try to make a duck radiation hybrid map. Ultimately, idea is to find out the sequences, and we know that you have made fused some 3 or 4 chromosomes. That means we know that chromosome 3, chromosome 4, chromosome 5, they have all been fused, and we are looking at specifically 2 or 3 chromosomes sequence, not other chromosomes, because we have made others 3. That is the purpose of making radiation hybrid. Whatever hybrid we want to make, hybrid between 1 and 2, we can make and we can study. Specifically, that means we focus on a particular portion, just the portion we are not doing. And a lot of dynamic arrays were used, integrated fluid circuits. I don't want to confuse further, but these are the techniques they are used to study the different sequence. If you look at that, how people are able to do mapping? Mapping essentially use intercrosses. What does it mean? You made crosses between two phenotypically or trait-wise two different birds. Here there is a mallard bird, a colorful duck that is the wild ancestor of the common day ducks. That is very tiny, very colorful, doesn't have a production. Hardly 20-30 eggs. Another side, there is a bird which is roughly around 3 kilo, 3.5 kilo plus adult, and it also lays around 200 eggs. This was not laying more than 30-40 eggs. This is laying 300 eggs. Let's say 200 plus. Khaki means 300 eggs. If you make a cross between these two evolutionary distant breeds, essentially they are both are ducks. 4 sequences are ducks. So what else is different is that the gene sequences, the minor genes and major genes, small QTLs, they have all been fused, defining a particular trait, and they have been conserved in a particular manner. In the mallards, they are not known for their productivity, but they are known to harvest the maximum amount of diversity in them. So whenever there is a study, people used to make crosses between two evolutionarily distant breeds. Here they made a crosses between the mallards and ducks and take the F1s. The F1s will look something like this. Intermediates. The F1s and F1s they are able to mate further, and they make a F2 population. What we also used, and Sivam also made some similar F2 process to study the difference between the Kadakanath and the white leg chickens. And though it's a different thing that where we reached, Indian technology permitted to reach us somewhere, and the purpose were different. But we are trying to identify how this FM gene segregates. Here, they do not know all these genes. What is the difference? Obvious difference, the size. And also they are trying to know the plumage color. White looking shiny green and shiny bright. Why this particular part is white? So if you just look at these, decipher which chromosome this particular color genes are located, which chromosome all these fatness and size genes are located, you have to essentially make a cross. and make F2s. F2s will be segregated. What are the F2s known for? They might be coming from the same parents. Let us say one pair of parents, one pair, just one mother, one father. And you have 20 or 30 or 40. So biologically 50% also can be done. All these 50s are going to be different in terms of segregation. One egg given today and that is given after 6 months, they are not going to be same in biological constitution because 50% it draws randomly from the father, 50% it draws randomly from the mother. And there lot of recombinations keeps going. That is how you know that how F2 is different from the F1. So if you study the F2 simultaneously using the markers and using the traits, then you are able to decipher with the modern day tools. Modern day markers is not the same markers what they used to have 20, 30 years before. RFLP marker, STR markers were used in early 90s or let us say late 90s, early 2000. Nowadays, we are not talking anything less than SNP chip. One SNP chip, that means theoretical, that is containing roughly 2 or 3 lakhs of markers in one bead. More than 6 lakhs marker Acumetrics is making. I'm going to show a picture there. So using that, they could make meaning out of this and they ultimately done. So I'll go quickly to cover rest of the slides. There are 3 main categories of marker systems. People all know that dominant, biallelic, codominant. Codominant means you are able to see both versions of the markers. See one, let us say dominant. Both markers can be seen together. That means that's a codominant marker. So whichever markers are codominant, they'll be more useful. Whichever dominant former, that means you are using a PCR and you are amplifying one fragment. That is basically dominant marker. What are dominant markers? Examples are your AFLPs, RAPDs. So I will not go into details of that because this entire slide I am sharing with people. So these are the different DNA markers used for markings. They are most often not coding sequences because they are going to lead us as the anchors. We try to use them saying that this marker is sitting here means this marker is linked to a body weight allele. That allele which is defining 2% of the total variation per se. And we are going to define it how? We cannot see because they are made up with QTLs, quantitative trait loci, all giving small effect, but they are located in a region where at a time 10, 20 or 50 loci may be giving rise to one significant QTL that inherits N block. How we are going to define? Look at the marker where the SLW 551 is going to segregate. Along with SLW 551, our QTL is going to segregate. To define a QTL, you have to use a marker to define that. So for that reason, people are using RFLP, SSLP, BNTR in the yesteryears, and these are the different properties of the DNA markers. How? From the RFLP days, we have come to SNPs, and these are the where SNPs. Why SNPs? I have stopped here because all the later day technology they are using SNP as the model. One chip they are going to put 2 lakhs SNPs, 6 lakh SNPs. Where from they have come? In chicken, they published around 3.3 million SNP way back in 2004. By now, they have sequenced the entire chicken genome, maybe 1,000 times or more. 100-fold, 100x sequencing is done. So they have now thread bare, they have researched all the chicken breeds. Same thing is happening in the dog circle, catching little later, but almost it has got there. So this is the earlier day mini satellites where the days, maybe you all remember Lalji Singh's name you might have heard, who solved the DNA fingerprinting, Naina Sahni cases in our younger days. Forensic cases they used to solve, and I had a chance to use the same Lalji Singh's probe, Alec Jeffrey's 33.6, 33.15, all these probes are used in human genome. Those probes are shared with us also. I tried to use my PhDs. So we have all seen that how you generate the DNA fingerprint. One individual looks from different, from third individual, or from fourth individual, sixth individual. All by virtue of these particular markers. And those days, radioisotopes are used. P32, P33 all were used to make this. Nowadays, they have become obsolete. RAPD markers were used in the early 90s. Even our students, Dr. Mahadev is there present. He will also agree that we made all our studies in the early 2000 using RAPD markers. They are again dominant markers system. They behave like markers, but you would never get to know which is a codominant, which is a recessive heterozygote versus the homozygous, because they will look all the same. One PCR primer will give an amplification. It doesn't tell you whether it is having a codominant marker in that or a dominant marker. Homozygote versus heterozygote difference is not possible. AFLP marker. This is also another dominant marker system. They use the principle of restriction, fragment polymorphism, and along with that selective PCRs. I have written all these things, you can read it later. And the latest kind of marker is copy number variation. Principle is same. Repeat unit maybe in thousands of base pairs, and copy number variation is the latest kind of markers because it's easy to get a bigger block being used as a marker. unlike the microsatellites, where 2 base, 3 base repeats will be there. TG repeats will be there. TG repeats will be baffling. Throughout the genome, TG repeats are there abundant, lakhs and lakhs. But if you talk of CNVs, they will be maximum in thousands. TG to handle thousands, then the lakhs. And ultimately, what they are making use of, they are making marker-assisted selection. They are making to make use of. And what the marker-assisted selection was doing, it is an indirect selection where the trait of the interest is selected not based on the trait itself, but on a marker linked to the. That means if I have to select a high body weight bird, I'm not going to weigh the bird. Physical weighing, I'm not going to. I am told by my guide or my co-workers, Dr. Mishra, these are the markers, marker X, marker Y, marker Z1, Z2, Z3, like that. I have 100 markers. How I nominate, that's up to me. A1, A5, A6. Wherever you find the bird which is positive for A15, G16, D32, and Z15, and let us say N35, all these 10 or 15 positive markers, that bird you are going to select. That means I will only do the laboratory study. Identify all the birds for the 15 set, what my colleague has told. I'm not going to look at the face of the bird also. I don't know. DNA sample have come to me by number wise. Number 1, number 2, number like that. Alphabetically, I might have number. I'm looking at the marker profile for which my colleague has said these are the markers which are meant for high body weight. I give a list out of the 300 sample given to me. These top 60 are these. Select them. That means I don't look at the bird and physically select the birds. And interestingly, they all become with high body weight. I don't have to bother. So for that, somebody must have proven that these markers are in quick linkage with the body weight genes. They become markers for me, and I make a marker assisted. Physical selection is not done. And what we are using, maybe microsatellite or maximum CNV or some marker we are going to use. Modern day markers have gone 1 step, 2 step, 10 steps higher. Not 10, 20s. पागल हो जाओगे फिर सिलेक्शन नहीं कर नहीं पाओगे. So somebody they say use this Apimetric gene array 60K array. My colleague Dr. Hans Singh was using in year 2008 itself. This is the SNP array. Look at it, Dr. Mishra. If this profile you find, select it. That means he has taken at a time 60,000 location and a particular profile out of the 60,000, 3,000 prominent marker profile. There is known as a. So batch 1, wherever you find out by search, identify that out of the 335 birds, 70 birds are matching positive for batch 1. Select that. Obviously, because they are based on many markers. Here we are talking of 15, 20 for lab. They are talking of thousands at a time. So they are making better, efficient selection. So accuracy of selection was there. All these years we have survived with selections only. All the breeders in the background, they do selection. For egg type, they make family selections, index selection. For meat type, they use mass selection. For simple body weight selection. Selection methods were there. Policies were known. Falconer was talking since 1930s. Procedures have changed. Understood? So this is all about the genome-wide marker-assisted selection. This is the latest selection that is happening with the chips. What I said. This is the version of gene chip. A machine that is there, and we're all going to put only one DNA sample on this. It is going to give information about 6 lakhs locations on that particular bird, about that particular bird, and do whatever want to interpret and do that. We'll quickly move towards a little bit of doc embryology. Why doc embryology? Because entire applications now has gone from application. This genomic knowledge we are going to with the transgenic chickens. You might have heard, no? What is transgenic? That means our desired modifications we are going to introduce into a particular chicken without going to select for it. But we want a chicken that is a Pekin chicken, Pekin bird, but we want this beautiful color of the blood. Can we take it there? We wish that our Pekins is big in size, big in production, big in eggs, but has the entire color sequence of the mallards. If I make a sequence and that process, it will take me a lifetime, and still I will not get have both the things. Mallard like feathers and the portion of what is that? Pekins. If I make it for transgenics, pick up 1 or 2 genes, try to introduce through the procedures, I get everything. That is the beauty of transgenics. Selectively use a foreign gene to be expressed in a recipient host. How people have done? People have traditionally used, and doc embryology important is that because entire duck genomes conservation, genomes biobanking, everything is done. Unlike mammals, where you can access the zygote, ducks you can access the zygote. The moment you are getting an egg, where you get? The egg is laid just now, 5 minutes before. You go and look at it. There is no zygote inside. It is already differentiated within the uterus itself. what I get is around 10,000 plus cell stage. Stage X has already arrived. So every time I talk of manipulating the chicken genome, I have to manipulate the primordial germ cells, or ectodermal cells, or some other cells, or the embryonic stem cells. All this. So based on this is a slide that is showing a how duck you can sex using a day-old sexing is a male. Look like this at day-old stage. First has, and my colleague Dr. Adnan Naim, who is also participant in this, also contributed to making these slides. He shared with me beautiful slides. Students may look at it. Day-old stage, if you look at it efficiently, you can sex a male from the female. No need to go for the conversion technology of using. Though molecular genomics help you to identify a male from the female by looking at the molecular profile, you don't need to do that. You don't need to spend that 1,000 rupees. These small techniques are always there. Where that is not feasible, do this molecule. Drug genesis, people have done a lot of work, and mainly the hosts are the blastodermal cells or the primordial germ cells. And techniques like CRISPR-Cas9, then we are going to have a vector to which you are going to try use your modifications, make a clone of that, and that entire vector you are going to pass into such cells which are able to transmit the entire genetic information from one host to the other. So PGCs have been long used for making these modifications. So duck genome biobanking, Dr. Adnan, and he was there with me for a long time, 2 to 3 years, and he made some beautiful work. And these all things is only 1 minute I'll spend on this. He is trying to use duck as a surrogate host, where he can make chicken-specific genes being expressed there. What you are able to see, a Kadaknath chicken. Why Kadaknath chicken is known? Dr. Shivam knows that. Kadaknath chicken has uniquely, I need just 5 minutes to wind up. So Kadaknath chickens uniquely they have a black gene in that. Means entire skin is black, the feather is black, the blood is black, the face is black, intensely black over pigmentation is there in the entire meat. People behave it differently. People, the tribals, they believe that only the goddess will be pleased if you sacrifice the Kadaknath. But we know that is because of hyperpigmentation, more proteins come into that. And more qualitative elements what Kadaknath is known for, that comes from that. We wanted to use Kadaknath as a, let us say we want to conserve Kadaknath. How we are going to conserve Kadaknath? To make a population of Kadaknath, 1,500 bars you have to go into. Every time you are going to maintain, then only you conserve. But physically you don't need to conserve like that. Take the Kadaknath's primordial germ cells out of the Kadaknath, and Kadaknath may be vanished after, let us say, 40-50 years. People know that Kadaknath doesn't have productivity. One fine day, Kadaknath might have vanished. It is now limited to Jhabwa. Government of India is making a lot of effort. But still, you want to ensure that Kadaknath is also conserved in my lab, in the fridge. That means I efficiently conserve Kadaknath in my fridge. The duck germ cells, duck genome, I conserve Kadaknath, and vice versa. Rare ducks also can be conserved in the chicken genome. In the white leghorns are rampant everywhere. Here, UK, Hungary, Japan, everywhere white leghorn is there. And my rare breeds, Kadaknath can be conserved either in a chicken cell or even in duck cells. Why duck cells I'm saying? Because ducks are available as a waterfall. So across species, also we can conserve these cells. And how people are able to do? Donor Kadaknath, take 2.5-day-old embryo, from that 6.5-day-old embryo, different stages you can harvest the PGCs, culture them, characterize them, identify which are the PGCs, and put them into cryopreservations. In the minus 196 degree, that much low temperature you put it, and in that the PGCs are safe for years and eternity. After 40 years, 50 years, when I'm too much old, I'm asked to revive Kadaknath, I can safely do it through this particular technique that is called chimera. A chimera, that means physically looking like a duck, but it may be hosting the Kadaknath's gonads, spermatozoa inside. That's efficient. This chimera technology has been done by Dr. Adnan and group for the first time in India, and is going to shortly make available. This particular diagram will explain you everything. I'm going to share with you. And there is some 1 or 2 small sequences, one measurement. I will tell you how it is done. Beautiful. Let's look at it. All the boy is harvesting blood from a 2.5-day-old embryo with a small microscopically. He is able to, very micro pipette, micro needle. He is going to harvest the 2.5-day-old embryo from that. When the heart is beating and the embryo is just like a spider, he is harvesting few microliter blood out of that, which will contain the potential PGCs, which can be cultured and be stored in our lab for eternity. That means Kadaknath is efficiently stored in my lab for thousands and thousands of years if you want. Technology has to be perfected, done elsewhere in the world, but done first time in India. And what is showing here in redness is only virtually 2 or 3 microliters. is being shown in a stereo microscope zoom. So you are able to see such a big needle there, but actually this is only 2 to 3 microns, maximum 4 to 5 microns. And through that enough PGCs come for the purpose. Okay, and you have to take out after that also the embryo should be live. That's the beauty of the technology. Okay, so we should give a big hand to Adnan that he has done it first time with little support with his money, whatever he got. And after he left, I think we have stopped that work also. So he is able to do from the gonads that, because in that stage you don't know, 2.5 days stage, we don't know male PGC and female PGC. But if he is using a embryo of 4.5 day old, he can know how he is able to harvest. He is able to harvest a testis out of it. Same way he is harvesting under the stereo microscope, and that testis is having the PGC inside. Okay. Here he is injecting back. If you make any modifications, genome editing, you are going to inject back into the embryo. Just look at it, how he is taking quantity of such PGCs and let us say edited PGCs and going to put back into the growing embryo at a similar stage. I mean, 2.5 day old embryo was used to harvest the PGCs. He is going to inject the same PGCs modified or whatever is PGC or Kadaknath PGC into the recipient in the same manner. Same micro needles and done efficient microscope so that they are done like that. So these are the characterized PGCs. You can differentiate. Time is not there to describe in details, but these are the PGCs versus the rest of the. And he has done different stainings to know that stage specific antigens are there. For periodic assisted stain cultures, he has made. He has made SSCA1 stained PGCs here, and he has also intraspecies chimeras he has generated, and he is conserving these PGCs for decades. And maybe after we retire, also still Kadaknath will be stored within the duck genome, and those PGCs will be there. And he has filed a patent recently. And let me conclude in under 1 minute. As a whole, ducks as a commercial importance poultry species also subject to biotech research at par with chickens, turkeys, guinea fowls. Everywhere research is happening, but we are happy to say that duck is not back. I mean, too far behind. They have done in 2004. We have sequenced in 2014, and duck genome has already been sequenced with sequence data, and polymorphism of common breeds of duck has been deciphered. Different duck breeds. Duck genome size is now known 1.1419 GB. That is Pekins, and 1.21 billion base pairs gigabytes. That is in Zhaozing duck, and 1.21 in the mallards. Mallards are always more because they are the evolutionary conserved genes there. Existing data in turn are the starting points to fuel applied genomics. If you know the baseline, if you know the inherent content of the duck genome, you do any modifications. You correct a portion, you remove a portion, insert a portion, delete a portion, make all the modification, all the techniques, upcoming techniques, use that, but basically you should have the draft. The draft is ready, everything you can do over that. Applied platform like PGC biobanking, embryonic stem cell technology, across the species gene transfer, industrial applications, they are going to be done on that, and ducks research will be current hotspots in biotechnology. Pace of generating DNA markers and QTL discovery are on the same footing like chickens, because who will be doing? The interested partners. This obviously things are going to happen more in China, because most of the ducks are there in China, but not need specific. And duck genomic selection is whatever has been practiced in Highline and other Abhiyajan groups in USA in chickens. Same thing is happening in this part of the world, in the Middle East, using the duck. And what are the requirements? Same SNP arrays. So SNP arrays for chickens have been developed since 2010. Here things are available now. Recently, 2016-17, people have made SNP arrays that can also be made available to duck researchers. So a high density SNP array profiling platform that should develop, and we should be not asked to pay in how much SNP array type may cost, maybe roughly 200 to 250 rupees as boiled down to. Earlier it was in thousands, 8,000, 7,000 rupees per single sampling. It has come down to hardly less than 500 rupees. Somebody said that 250, 260 rupees on typing cost can be there. If that is happening, means we are going to do rest of our selections in India to this. Wish that arrives early and we are able to use molecular breeding. Whatever you want to do, we do molecular way. That means physical also, and use this molecular tools so that your accuracy is foolproof. Foolproof. Like DNA fingerprinting is the ultimate test for paternity. There is no doubt thereafter. A suspected baby is only known by DNA fingerprinting, because that is accuracy. So if you are going to selection, tomorrow's selection by molecular breeding, that is also accuracy. So tomorrow's, next year's, next decade's dimensions will be through molecular. I introduced ducks in the context. And thank you very much for your patient hearing. If anybody has a question, can ask. Else, I'm going to share this slide with all of you. So I thank you all of you for patiently listening, and I thank the organizers, RFI, Christian College of Eminence, and Madam Dubeji, Dr. Shivam Pratap, all people for giving me this chance, and happy that we are discussing and will be available in future also.
Thank you very much, sir, for such a enlightening speech and presentation on advances in genomics and biotechnology of ducks, the next important alternate poultry. It's really beneficial for us. Thanks once again. Now,
you.
welcome, sir. Now, one more eminent personality is among us. Yes, respected Dr. Supriya Kumar is among us. She is international speaker, communication and leadership coach, soft skill trainer, global motivational and keynote speakers. She is also social activist, chairperson of Positive Thoughts Counseling and Training Solutions. She is a training professional with over 15 years of rich global experience in setting up, leading and managing end-to-end learning and organization development management. She brings extensive experience in building programs for the leadership development pipeline based on business plans and has the expertise in providing ILTE based and blended learning solutions. Her facilitation and delivery capability, including corporates, L and OD MDP, LDP, and LDP programs, gamification, career development, ADDIE, content management, mentoring and coaching, communication and presentations, performance management, and process enhancement. She is awarded with various national international accolades like Best Soft Skill Trainer and Keynote Speaker by Corporate School World Summit and Awards, Best Speaker by Educus Unite of Scholar. Welcome, ma'am, once again. And now I requesting Supriya ma'am, please share your knowledge with us. Over to Supriya ma'am.
I hope everybody can hear me.
Yes, ma'am. You are audible.
Yes. Well, a great afternoon, everyone. And thank you so much, ma'am, for giving me such a beautiful introduction. And of course, once again, thank you so much, Dr. Ajay Jain, for giving me this beautiful and brilliant opportunity on this international conference on challenges and prospects of biotechnology environment. And trust me, folks, biotechnology is my passion where I diversified into a soft skill trainer. So you can see it's my love, first love, you can say, biotechnology. So thank you so much and hope you all can see my screen.
Yes, ma'am.
Yes.
Okay. Well, I'm not able to do the full screening, some technical glitches. Well, let's take it off. All right. When I use the term biotechnology, everybody is aware it's the fastest growing, you can say, the field in national and international places, where it is an amalgamation of biology, engineering, sciences, technology, and it is using biological system or its path to develop useful substances for the mankind. It is not at all, an unaware fact with anyone. And we all know the importance of biotechnology in today's era. If we talk about India, if we talk about global, we know what are the uses we are getting, what are the benefits we are getting benefited from biotechnology. And if I say about agricultural biotechnology, so we can say agricultural biotechnology of any country gives a boon. You can say a boon to the society, boon to the humanity, because agriculture is everything for any developing country, giving prospects, giving prosperity to the people, of having income, of youth employment, and of course, the stocks, the breeds. What we are having today is all the boon and gifts of agricultural biotechnology. And hence, we can say how important it is. If we see the global status of biotech crops in 2005, we can analyze how important and how we can say without agricultural biotechnology, we cannot think the prosperity of any country, and hence it lies its importance. And it is very plays a vital role in maintaining a country economic prosperity, stability, by meeting the growing needs of food, income, and of course, as I said, youth employment. So with this graph, with this status, with this biotech crops, we can say the importance is immense. And what are the scientists, researchers are doing in this field is immeasurable, and it is immense. Well, if I sum up, or I will show, the agricultural biotechnology is the collection of scientific techniques for improving plants, animals, and microorganisms based on understanding of DNA. And what the scientists are doing, and they're finding n number of solutions. They are working day and night very hard to discover new products, benefit for mankind, benefit for a society, and of course, benefits for the agricultural economy. And that's where we see a change. And we should be thankful to all the scientists, and a lot of gratitude and respect for increasing our agricultural productivity, enhancing breeders' ability, and improvement that are not at all possible without them. So you can see n number of helpful, meaningful things which they are discovering day and night. And before I take up this topic of challenges, let me apprise you all with its benefits. And trust me, folks, benefits are immeasurable. That is, you can say, with the help of this agricultural biotechnology, we have increased crop productivity, crop production enhancement, nutritional value of crop getting improved day by day in the market, whether it's an Indian market or global market. And of course, chemical herbicides avoiding is always seeing a graph reduction when we are comparing virus resistance and delayed fruit ripenings. So these are the benefits which we can say we can look here. But apart from these benefits, we have n number of advantages that we are, getting prospects with the help of agricultural biotechnology. And all credit goes to our scientists who are working day and night hard in this industry. Well, now, in every challenges, we have an hidden opportunity. And if I talk about the challenges in agricultural biotechnology, so let me be very realistic from my own experiences. When I was in my biotechnology MBA, I seriously found that there is a poor funding which I faced in my laboratory. We cannot buy a few of the equipments, few of the substances which is necessary to conduct any research. And from my own experiences, I have collected these challenges which I am presenting in front of you, all the eminent personalities. Lack of effective leadership is there, but most important thing is poor funding, which will, however you are talented, however you are genius, how you are brilliant and knowledgeable in your expertise area, But until or unless you have a good funding, you can't prosper. You have to look for other options. If good funding is there, you can buy your equipments, you can buy your laboratory things, substances, and you can carry out your research in a very successful and a very lucid manner. But funding is the part where we are lacking, and we blame sometimes government, we blame sometimes our university not to provide us. So this is the core area where I face challenges in my time when I was working in my laboratory. And of course, this is one of the reasons why I think most of the youngster scientists, they are leaving India and moving abroad. So this key area, if we will improve, definitely. And of course, I can say 100% guarantee I can give you, we can preserve our scientists in our own country. So let's remove this, take up this challenge, fill the gap where we are inadequate funding of agricultural biotechnology. And this is one of the main challenge which a person, or you can say a normal person, or a layman or a scientist usually facing. And the graph, the survey has shown this is to be the 100% reason. And of course, your lack of research focus, infrastructure, inadequate human resources, and expertise area is always there. So we can't say we should be away from the challenges, but challenges, let's find the gaps, let's fill it up with the perfect solution. And of course, that time we will not find any challenges or any gaps in the field of research, in the field of agricultural biotechnology, and we can happily carry out our research. So these are the challenges which I always want to focus every scientist, every eminent personality who are sitting over here. Well, I will not take much of the time because all people are also there. So I will say only one thing, that every benefits is directly proportional to your challenges. If we are talking about agricultural biotechnology prosperity, utilization, we have to balance challenges and our benefits together. And then only we can flourish agricultural biotechnology. And of course, I will sum up by saying only one thing, that there is no place like our Mother Earth. Let's preserve it, let's conserve it, let's safeguard it, and let's make it a better place rather than polluting, rather than finding problems. Let's focus on solutions and make it the best place to live in. So it's my short presentation which I have prepared, seeing my time limit. Thank you so much once again for giving me this wonderful opportunity and sharing a great knowledge with everybody. Thank you so much.
Thank you very much, ma'am, for your impressive presentation. You focused on agriculture, biotechnology, challenges and concept also. Thank you once again, ma'am. Now, I feel great privilege and honor to introduce today's eminent speaker, and Dr. Sudhakar Umlesar, Head and Associate Professor, Mechanical Engineering Department, Sardar Patel College of Engineering, Andheri, Mumbai. His areas of research are IC engine, heat transfer, automobile engineering, ductile cast iron, biofuels. Sir guided 40 students of ME, MTech, and PhD. Dr. Umle delivered expert lectures in FDPs, STTPs. He conducted and coordinated courses in SPCE, FDPs, STTPs. His membership of scientific and professional societies. He is life member of ISTE, ISNT, and IET, and many more. Sir has technical judge, session chair, reviewer, editorial board member, and organizing committee member in technical events, national and international conferences. Sir has been the recipient of many awards like Best Technical Paper Award 2015 by Indian Institute of Foundrymen, SAEI and India Guru Award, and many more. Welcome you, sir, wholeheartedly. And now I invite you, sir, please share your knowledge with us. Sudhakar,
Yep.
sir?
Am I audible, madam?
Yes, sir, you are audible.
So
Over to you, sir.
please give me the presentation authority.
Ajay, sir?
Huh. Okay. The PPT is visible.
Yes, sir, visible.
Yes. Dear participants, Dr. Ajay Jain, National Coordinator of Research Foundation of India, Prerna Dubey Madam, RFI Moderator, Chauhan Sir, Director of Eminent College of Christian, Dr. Saurabh Jain, Professor Ashok Gupta Sir, Professor Dr. Shiv Pratap Sir. Welcome you all in my today's lecture on agriculture technology in India. Why I have chosen this topic is that the agriculture sector is one of the major sectors in the Indian economy. According to the Economic Survey 2020 to 2021, GDP contribution by the agriculture sector is likely to be 19.9% in 2020 to 2021, increasing from 17.8% recorded in 2019 to 2020. Over the years, the government has taken major steps to add and enhance the agriculture sector with proven agricultural technologies and supportive policies. The recent evaluation of agriculture 4.0 technology in agriculture will further accelerate growth of higher crop yields and enhance sustainability by reducing water consumption and use of agrochemicals. Indian's agriculture scenario, agriculture plays a crucial role in the life of an economy. It is the backbone of our economic system. Agriculture not only provides food and raw materials, but also employment opportunities to a very large proportion of populations. Agriculture is the primary source of livelihood for about 58% of Indians' populations. This is very important. You must know what is the role of agriculture in the economic development sector. Agriculture sector plays a strategic role in the process of economic development of the country. The agriculture sector is the backbone of an economy which provides the basic ingredients to mankind. The products of the agriculture now become the raw material for industrializations. In India, the majority of income comes from the agriculture sector. You know all. If agriculture fails to meet the rising demand for food, then the growth rate of economy suddenly affects. Therefore, agriculture advancement is necessary for improving the supply of raw materials for agro-based industries. And hence, I have decided to give and concentrate on agriculture 4.0 technology. So, agriculture revolution is very important to this agriculture area so that we can get the more yield. The agriculture revolution began in Great Britain around the turn of 18th century. Later on, we have so many revolutions. Now we have the fourth revolution, and hence we are calling it as agriculture 4.0. The agriculture revolution is necessary for the growth and improvement for the farming and agriculture industries. Hence, it is important to understand causes, inventions, effects of new farming techniques. And hence, I will cover all these causes, invention, and effects in my lectures. The necessity of agriculture revolution is required to increase agricultural production by factors like the increased availability of farmland, a favorable climate, more livestock, and improved crop yield. So we have to discuss about what are the different components required for fourth agriculture revolutions. This period saw the development of crop rotations, technical improvements to the crop to make it more efficient, and gradual increase in farm size. Farm products began to be traded nationally, and farms became more established as a business. And hence, when you want to turn agriculture into business, you have to observe so many things like industries, and hence farms as well as farmers are very important. So what about farmers? Farmers must be given proper training to run the agriculture 4.0. And what is the agriculture role in India? It is very important. And hence, there are so many revolutions in this agriculture revolutions in India. What it means? Agriculture revolution refers to the significant changes in agriculture when there are inventions, discoveries, and or new technologies implementations. And these are three are very important for the development of Indian agriculture. That is, first is inventions, discoveries, and new technologies. These revolutions change the ways of production and increase the production rate. Various agricultural revolutions have occurred in India and have marked the beginning of a completely new era in the agriculture field. The agriculture revolutions helped Indian agriculture grow exponentially and created new opportunities. So, what is the need of research in agriculture? It is very important. As our topic is agriculture research, we have to concentrate on what is the importance of research in agriculture. And hence, the primary role of agriculture research is to enhance knowledge and improve technology. Research in agriculture is required for improving productivity and quality of crops. Research is required in area of agricultures like better plant protection, irrigation, storage methods, farm mechanizations, efficient marketing, management of resources. And hence, I will cover all these protection, irrigation, storage methods, farm mechanizations, efficient marketing in detail. And hence, we for the research we require the agriculture educations. And what is the need of agriculture education for the betterment of Indian agricultural systems? And hence, agricultural education is defined as a systematic program of instructions for students desiring to learn and educate around the science, business, and technology of agriculture, food and environmental, and natural resources systems. Agricultural education prepares students for success careers and informed choices regarding agriculture. And hence, at the end of my lecture, you can understand what are the careers in agriculture sector so that we get lot of research through the education. And hence, what is the need of research in development? So already we require the development in the agriculture, and need of agriculture research is required for the growth in the agricultural productivity. Research gives ways to increase farmers' profits and to protect environment. Different technologies used in agriculture like sensors to identify soil moisture, smart water systems, and precision farming using geospatial technology. These are very important. Investments in agriculture are the best weapons against hunger and poverty, and they have made life better for billions of people. So this is a very important hunger and poverty. Poverty is a very serious problem in Indian scenario. So only agriculture development can reduce poverty as well as hunger, and hence we require the agriculture development through industrializations of the agriculture field. We require the technology, and that technology is called as Industrial 4.0. And hence we must understand exactly what is agriculture 4.0. The agriculture 4.0 technologies such as artificial intelligence, shortly called as AI, machine learning, shortly called as ML, remote sensing by big data, blockchain technology, IoT, Internet of Things, are transforming agricultural value, modernizing operations. Still in India, adaptation of these technologies are not possible and not implemented. But if you start thinking and researching on these areas, definitely the agriculture field will grow, and hence we can say that this is nothing but agriculture 4.0 is nothing but fourth agricultural revolution, and this fourth agricultural revolution is very similar to the fourth industrial revolution. It is called as industry 4.0. This agriculture 4.0 refers to the anticipated changes from new technologies, particularly the use of AI to make smarter planning decisions and power autonomous robots. So you know that today we are observing autonomous vehicles. And there is no driver to run the vehicle. Similarly, it is necessary to think on what are the developments we do in robot developments, and hence there should not be any type of autonomous there should not be any type of man made or man drive the robots, and hence completely autonomous robots we require in the agriculture field. In September 2021, the Union Minister of Agriculture and Farmers Welfare, Mr. Narendra Singh Tomar, announced the initiation of the Digital Agriculture Mission 2021 to 25. And hence, Agriculture 4.0 is a term for the next big trends facing the industries, including greater focus on precision agriculture, the Internet of Things, and the use of big data to drive greater business efficiency in the face of rising populations and climate change. The rising population is very serious in Indian scenario. We have to think about how the food is produced and supply of food to the all populations. Similarly, we are facing a lot of challenges in changing climate, and hence we have to consider the climate change scenario for the development of agriculture through industry as well as agriculture 4.0. And in 2018, the World Government Summit published their report called Agriculture 4.0, that is the future of farming technology. The Agriculture 4.0 incorporates the evaluation of precision farming and refers to all actions that are carried out in agriculture based on precise and accurate analysis of data and information collected and transmitted through advanced tools and technology. The aim of this technology is to offer the most extensive and precise support to farmers in the decision-making process related to their activity and the relationship with other parties in the supply chain. And you must understand adapting 4.0 solutions in agriculture means. So like, what is exactly how we implement this agriculture 4.0? The ultimate goal is to increase economic, environmental, and social sustainability, as well as profitability of agricultural processes. So through this agriculture 4.0, we are focusing on increasing economy, environmental conditions, and social sustainability. Agriculture 4.0 refers to the use of Internet of Things, big data, artificial intelligence, robotics to extend, speed up, and increase the efficiency of activities that affect entire production chain. Avoiding unnecessary waste by calculating exact water requirements of the crop or detecting in advance the onset of certain plant disease or pest. Having greater control over cost and being able to plan all stages of cultivation, sowing, and harvesting with great deal of precision, saving both time and money. And this is very important that what is the future of agriculture? And hence I have given the question marks, future agriculture. So today we are observing what is agriculture conditions, and after 20 years, what would be the new agriculture scenario. And hence I used future agriculture. Future agriculture will use sophisticated technologies such as robots, temperature and moisture sensors, aerial images, and GPS technology. These advanced devices and precision agriculture and robotic systems will allow farms to more profitable, efficient, safe, and environment friendly situations. The future of agriculture cannot be achieved without digital tools and a strong thrust from technological innovations. This is very important. We require first thrust for the innovations, and through this innovation, we do the appropriate research for the agriculture 4.0. This is very important now that is the actual involvement of science and technology in the agriculture development. So what is the need exactly? And hence there is an urgent need to identify the different problems and their solutions in agriculture sector. So what is important in the system, like when we do the PhD, we are identifying so many different problem statements. Through that problem statements, we do the solutions of that problem statements, and after getting solution, definitely we get the PhD. So if you apply same systems in the development of agriculture, what are the problems, collect your problems and solve the all problems through technology. Science and technology has started to find out the solution in the agriculture sector. The Indian Council of Agriculture Research is engaged in harnessing modern scientific knowledge to develop high-yielding, open pollinated varieties and hybrids that are resistant, tolerant to major biotic, abiotic risk factors, efficient input use, and adapted to different agro climates suited for irrigated and rain-fed areas. So research is very important, and you have to find out the question that is need of research in new technology. So why we need new technology already I have explained, and what is the research position for that new technology, I am giving the answer of it. We require advancement in agriculture sector like industries to develop agriculture field. In Indian scenario, we have a drawbacks in agriculture developments, and when we want to make it like industries, in industries you know that they are using so many technologies for the generation or manufacture of the product. Similarly, in agriculture sensors, devices, machines, and information technology in agriculture. It is also necessary to develop technologies like robots, temperature and moisture sensor, GPS technology. And hence, we have to understand first what is the importance of agriculture technology in the development. Development of agriculture technology is necessary in basic requirements of farm like water supply, fertilizers, and pesticides. These requirements may be required in the minimum quantities in such a way that the farmers would get more profits. Basic research in agricultural technology. So you must understand what is the exact basic research in agricultural technology. So the research is required to improve crop productivity, minimum use of water, fertilizers and pesticides. Those are helpful in reducing food prices down. So this is very important. The food may be available in plenty for the populations, and hence the cost of the food may be reduced when we have a very sophisticated technology. And for that we require the education that way, and hence how to provide the education to the students, research scholars, postgraduate students in the agriculture field. And hence also we have to provide some training to the farmers, because if the farmers are illiterate, they cannot run protobots, or they cannot use drones, they cannot use this advanced technology without knowledge. And hence, because these are the challenges in front of farmers, and hence they have to give the proper training to understand the complex challenges in agriculture area. During implementing scientific and recent technologies, farmers must be trained to use these technologies. Even also to develop and improve these technologies, research must be undertaken. To promote research, teaching and research engagement would be promoted through research scholars and through grants for scholarships and fellowships. So this is very important. Government of India has started already scholarships and fellowships for the students for the development of research. And this is very important. How do you give face the challenges, national and international levels? So these challenges are very important for the Indian scenario. How to improve the, how to enhance all this technology, and we need help from the national and international level, and hence we have to develop the cooperation from different countries for the development of new technology in Indian scenario. And hence what is the role of modern technology in agriculture is, it is also important. Agriculture in India is largely dependent on nature, but climate and global warming issues make farming unpredictable. The need of the hour is to educate farmers in the use of modern technology and innovate approaches to increase productivity and raise profitability. Technology has a major role in farming and agriculture practices, and with the advent of digital technology, the scope has widened, and hence we will concentrate on digital technology in agriculture. And hence a role of modern technology in agriculture is very important. The basic of this is innovation. We have to concentrate on innovation. Innovation in agricultural is leading an evaluation of agricultural practices, thereby that reduce losses and increase efficiency. This is positively impacting farmers. Use of digital and analytical tools is driving continuous improvement in agriculture, and the trend is here to say, resulting in improving crop yields and helping increasing the income of farming community. And hence we must understand what are the importances in agriculture. Technology in agriculture affects many areas of agriculture, such as fertilizers, pesticides, seed technology, etc. Biotechnology and genetic engineering have resulted in pest resistance and increased crop yields. Mechanization has led to efficient tilling, harvesting, and reduction in manual labor. Irrigation methods and transportation systems have improved. Processing machinery has reduced wastage, and the effect is visible in all areas. And hence, we require the technological advancement, improved productivity from mechanization of agriculture to reduce manual labor and make the process faster. Combined harvesters are finding greater use, climate and weather predictions through artificial intelligence, modern equipment and tools based on artificial intelligence. Drones, remote sensors, and satellites gather 24 by 7 data on weather patterns in and around the fields, providing farmers with vital information on temperature, rainfall, soil, and humidity. And hence, we have to do the very recent agricultural practices. Agriculture practices mean basically a collection of principles to apply for farm production processes in order to get better agricultural products. They are simply practices used in agriculture to facilitate farming. It helps to create food for consumers as well as for safe further processing. Our farmers are the key persons for the cultivation of crops to provide food to the entire population. I have to explain some of the challenges. I will complete in 10 minutes. Some of the challenges of using agriculture 4.0. So we'll discuss all the challenges, and I appeal to all researchers. You would be able to solve these challenges. So this is one of the challenges, how to use the artificial intelligence in the agriculture. And hence, artificial intelligence is new technology to the farmers, and hence improve this overall harvest quality and accuracy, which is known as precision agriculture. Artificial intelligence technology helps in detecting disease in plants, pest, poor nutrition of farms. Yeah, artificial intelligence sensors can detect target weeds and then decide which herbicides to apply within the regions. Artificial intelligence technology that instructs machines to assess specific situations and make decisions in real time. The accumulation and ability to process and interpret large amounts of data is the main input for machine learning. Robotics using machines that automate certain tasks. Now, another technology we are using, that is a smartphone. So everybody is using smartphone for any other purpose. Now, this is our challenge, how to use smartphone technology in the development of agriculture. Smartphones have become useful tool in agriculture because their mobility matches the nature of farming. The cost of the device is highly accessible, and their computing power allows variety of practical applications to be created. Now, another challenge in front of researcher is how to develop Internet of Things in the agriculture and how to use IoT. The IoT has seen a huge acceleration in recent years, with smart devices becoming more prevalent and increasingly able to share with one another. The sensors on farm are already used to monitor soil, nutrition, temperature, moisture, and more. IoT means connecting all those systems together, removing the need to repeatedly enter data into multiple apps that don't talk to one another. This technology enables a variety of tools, for example, drones, sensors, or satellites, to connect, communicate with other in order to exchange information as well as data useful for improving crop development conditions. So these are very important. How to Another challenge in front of researchers is how to use drones and how to develop the drones technology. Now this is the latest advances in sensor technology, means drones are now able to use additional wavelengths in the light spectrum to assess crops, spotting weeds, and seek crops from the air. Drones are small unmanned aircraft that are able to monitor crops in real time as well as transmit images and useful informations. They are mainly used for land mapping, but the most advanced versions adopt infrared sensors and imagery systems to detect problems that cannot be detected by the naked eyes. Environmental sensors, on the other hand, are capable of recording climate weather data and information on soil moisture requirements. And uh, there are many challenges faced by the agriculture, particularly in India. So I will not explain all this. The first challenge is low productivity levels. Second challenge is underdeveloped irrigation facilities. Third is groundwater depletions. Fourth is lack of insurance support to the farmers. Next is lack of farm mechanization. This is very important. Farm mechanizations, already I have explained. The farm mechanizations through research, we can solve the problems. Next challenge is inadequate storage facilities in India. How to improve the adequate storage facilities so that farmer can store their crops, their foods. Now, this is very important challenge in the Indian scenario, that is agricultural market. Farmers are searching accurate marketplace, but it is not possible. There is a difficulty in supplying all the foods, all the vegetables, because actual markets are available far away from the place, and hence it is giving actual place, and hence if the researchers comes to give the solution for this, so that farmers can satisfy their activities. So another, it is very serious challenge in agriculture scenario, that is lack of soil health informations. So these farmers cannot get the idea about which food, which soil quality is required for food quality, and hence healthy soils must be provided, and farmers must be trained to identify the healthy soils and soil quality to improve the productivity of the crops. Another challenge in front of farmers is agriculture export policy. Government of India must concentrate on this export policy, and through that they can export Indian production and better farmer realization and synchronization within the policies and programs of Government of India. This is very important. We must develop some of the policies, and for that we require continuous innovations and efforts towards productivity, pre and post harvest management, processing and value addition. Use of technology and infrastructure creation is an imperative for Indian agricultures. Another challenge in front of the researcher is lack in food processing. This is the key challenges faced by the food processing sector. These are the gaps in supply chain infrastructure, which means inadequate primary processing, storage and distribution facilities, the insufficient connection between production and processings. This is very important, lack in technology development in the Indian scenario, and how our country is developing technology for the development of the agriculture field, and hence there should be drastic change in the innovations, and that agricultural innovation must be used for the development of agriculture 4.0 technology, and that can eliminate poverty, and hence it is helpful for rising demand for food and crops for the different climate change. This is very important. We have very lack research and development facilities in Indian scenario. Whenever you go, there is no research facilities or development facilities, and hence when we want to think about agriculture development, we require the a provision for agriculture development, and hence particular efficient R&D department we require for the improvements of the agriculture technology, for the improvement in productivity, and hence there is a reduction in pressure on the natural resources. Another challenge is the crop diversifications. So when we have a different crop diversification, we have a wider choice in the production of variety of crops. So this is very important. We must have the lot of diversification. We have a choices of crops, and the farmers must be trained to select their crops once in a year, or two crops in a year, or three crops in a year, and the rotation of the crops can be understood by the farmers. So for the growth of these crops, crop rotation must be understood, and hence crop diversification in the Indian perspective depending on crop pattern, considering irrigation, rainfall, and soil fertility. These three are very important. What is the irrigation facilities? What is the rainfall conditions? What is the soil fertility for a particular crop? And hence, understanding of crop diversification is very important. The country recorded impressive achievements in agriculture during 3 decades since the onset of Green Revolution in late 60s. This enabled the country to overcome the widespread hunger and starvation, achieve self-sufficiency in food, reduce poverty, and bring economic transformation in millions of rural families. Thank you very much. Thank you for patience listening. Thank you, Dr. Ajay Jain, for invitation as a speaker.
Thank you so much, sir, for your fruitful presentation on the future of agriculture technology in India. You focused on Indian agriculture scenario, role of agriculture in economic development, need of agriculture revolution, components of fourth agriculture revolution, and agriculture revolution in India, and many more. Thank you so much once again, sir. Now, respected Professor Dr. Pratik Rajan Mungekar are also among us. Welcome, sir. Dr. Pratik Rajan, sir, started his teaching at a very early age and has been working in the field of education and research as a professor, scientist, and counselor for 11 years. Taught more than 8,000 students and guided 4,000 students till date. Honored more than 650 state, national, and international awards for his contribution in the field of teaching and research. Received more than 245 honorary doctorates from all over the world and also sets 15 world records. Written many papers and articles in various international magazines, journals, and in books. Co-authored 7 books and written 1 book on sustainable development goals, which has got many international awards. He is visiting faculty in so many national, foreign universities. He is working as a professor and global educator, International Internship University, IIU, appointed as a president of Youth Development Council International Internship University. Participated as a keynote speaker in many international conferences, appointed as a planetary minister of sustainable development kingdom of Atlantis. Welcome wholeheartedly, sir. Now I request Mungekar sir, please share your knowledge with us.
Yes.
Over to Mungekar.
Thank you so much, Prerna Dubey ma'am, for your kind introduction.
Welcome, sir.
Good afternoon, one and all present here. I would like to thank Dr. Ajay Jain sir for inviting me here, for having me here. Deep gratitude. Now I will start with my presentation. So, Challenges and impacts of agricultural biotechnology on developing societies. Next slide, please. Hello. Next slide, please. Okay. So this is my introduction. Next slide, please. Okay. So the most critical areas in the world, this is abstract of my presentation, the most critical areas in the world for bringing economic prosperity and stability are the developing countries like India. Agricultural productivity in these countries must advance more rapidly to meet growing food demands and raise incomes while protecting the environment for future generations. Agriculture biotechnology has the potential to play a large role towards this achievement. Sadly, this opportunity remains a mirage for most developing societies because of numerous challenges that prevent them from benefiting from the uh, technology. This review identified these challenges to include lack of effective leadership, poor funding of agricultural biotechnology research and development, lack of research focus and infrastructure, and inadequate human resources and expertise. It further appraised the benefits and risks of agricultural biotechnology to developing societies. Among the potential benefits of the technology are an increase in the productivity of tropical commodities to meet future good needs, new opportunities for the use of marginal lands, and a reduction in the use of agrochemicals. However, associated with the technology are diverse questions of safety, ethics, and welfare. So this review concluded with recommendations on the appropriate use and application of agricultural biotechnology in developing societies. They include adequate regulatory measures, public debate, human resource development and training, public-private sector collaboration, intellectual property management, and support from international development organizations. Next slide, please. So, introduction. About 1.2 billion people live in poverty, earn, you can say, earn less than $1 a day, and majority, 70% of them live in rural areas. The outlook for developing societies in food and fiber production is particularly green. There are about 790 million undernourished people in developing countries whose food intake is insufficient to meet basic energy requirements on a continuous basis. Modern agriculture biotechnology has the potential to play a large role in more rapidly advancing agricultural productivity in developing countries. Transgenic crops represent promising technologies that can make a vital contribution to global food, feed, and fiber security. The creation of modern agricultural biotechnology involves input from basic scientists such as molecular and cell biologists and cytogeneticists who are not part of the traditional agricultural research extension network. Recent scientific advances in genetics have opened up a range of potential new applications of modern biotechnology. It shows that basic agricultural biotechnology research is costly and too demanding for scientific skills for the limited resources of most of the developing countries like India. This is as a result of numerous challenges, erroneous ideas, and beliefs which encumber research, development, and adoption of modern agricultural biotechnology in developing societies. Therefore, what are the challenges that hinder developing societies from embracing this innovation? What impact does the new technology have on them? And how then could agricultural biotechnology be used or applied so that the majority of the developing societies benefit from the technology? Next slide, please. Next slide, please. Is it visible, everyone? Hello.
Yeah, yes, sir.
Okay. Next slide, please, sir. Okay. Thank you. Challenges to agricultural biotechnology in developing societies. While agricultural biotechnology holds enormous promise for significantly increasing food production and relieving uh, already strained land and water resources, it has become an emotional issue among some consumers, environmental groups, and some societies. As the science continues to be developed, it clearly presents numerous challenges which hinder its development throughout the food chain, and these include, next slide, please. Okay, thank you. First, lack of effective leadership. The lack of effective leadership in science and technology and the absence of clear priorities, policies, and investment strategies in research and development (R&D) pose great challenge to agricultural biotechnology development in developing societies. The controversy about genetically modified GM food aid in many countries like Africa, India, for example, will continue to confuse policymakers and cause public anxiety if scientists and politicians in developing societies do not provide leadership to articulate their aspirations and interest in biotechnological development. Already, in number of countries, there is inconsistency in political uh, pronouncements and policies on biotechnology and its genetically modified products. The public is often left confused about contradictory positions and policies on the role of the technology in agriculture. Sometimes international political system influence political leaders' decision to reject proposals on biotechnology development. Second challenge is poor funding of agricultural biotechnology research and development, which is R&D. Biotechnology developments need high inputs of finance, which are in short supply in most developing nations. Even where the economy of such nations is somewhat stable, the budgetary allocation to science and technology and biotechnology R&D in particular is not encouraging. Much of the developing societies today depend on public sector investment in agriculture, research, and extension. But over the years, budgets for research and the quality of national research institutions have declined in many developing countries. Support from developed countries and donor agencies are declining, and there has been a tendency to spread investment in research and extension over a large number of institutions rather than developing a few quality ones. Next challenge is lack of research focus on infrastructure. Most developing nations have not identified specific areas of modern agriculture biotechnology in which to invest to meet specific goals. In the absence of identified national priorities, it is difficult to make informed long-term policies. Many developing countries tend to spread their limited financial and human resources thinly across diverse agricultural biotechnology activities and research agencies. While many have recognized the importance of setting biotechnology priorities and consolidating resources in a few research institutions that have the potential of growing quickly in centers of excellence in biotechnology. The countries have not established and applied strategies of identifying such institutions and ways of setting priorities. They continue to operate with isolated, completing, and often scientifically weak research agencies. Such agencies lack scientific and technological infrastructure for sustained agricultural biotechnology research and development. Next challenge is inadequate human resources and expertise. Agricultural biotechnology is an intensive research. Can you show the previous slide? I think you have. Hello. I would like to request the technical team. I'm on the challenges to agriculture biotechnology in developing societies. I guess fourth slide. Hello. Can you show us the fourth slide?
Please go on fourth slide. Technical team, please.
Now my PPT is not visible. I think they.
Right.
Yeah.
Yes, sir. Hello, technical team.
Okay. I'll tell you when I want the next slide. So please go on fourth slide. Yes. Challenges to agricultural biotechnology in developing societies. Next slide. Yes, right. Thank you. So inadequate human resources and expertise. Agricultural biotechnology is an intensive research area which needs high capacity of human resources to achieve substantial benefits. Most developing societies lack this human capacity where they are available. It is in short supply or they are not given enough opportunities to express themselves. This results in their drifting away to the already developed societies where their knowledge is valued, a phenomena oddly referred to as brain drain. The low level of scientific literacy in developing societies does mean that most people will not be able to draw informed conclusions about important agricultural biotechnology issues. Consequently, it is conceivable that a small number of activists can argue the case against generic engineering in such emotive and ill-reasoned ways that the public and politicians are involved. The general public and farmers in particular are not informed about the nature of the technology, its potential benefits and risks, and rarely do they participate in deciding what crops or problems agriculture biotechnology research and development should focus on. Agricultural extension has a great role to play here through mounting of awareness campaigns on biotechnology product information, disseminating proven and reliable biotechnology products, involving end users to participate in issues relating to biotechnology R&D, and supporting end users to adopt the new innovation in order to achieve food security. Please, next slide. Next slide, please. Impacts of agricultural biotechnology on developing societies. There is no doubt that genetic engineering, GE, has the highest profile among the new technologies in agriculture today. Transgenic crops like maize, rice, wheat, soybean, and cotton are among the top priorities for the agricultural biotechnology industries. This, notwithstanding, GE also has created the strongest sense of unease and resistance among consumers, developing country farmers, and environmentalists. While there are undoubted benefits from the use of transgenic organisms, it is important to ensure that such systems do not cause problems of safety to people and the environment or create unacceptable economic, social, moral, and ethical issues. The likely benefits of agriculture biotechnology in developing societies are an increase in the productivity of tropical commodities to meet future food needs, new opportunities for the use of marginal lands, and a reduction in the use of agrochemicals, production efficiencies, and bigger yields provided by the agricultural biotechnology may bring down prices for rural, poor, and urban consumers in the long run. Also, genetically GE genetic engineering is increasingly applied to many breeding programs to achieve the same aims as the traditional methods, but offering two main advantages. First, the introduction of genes can be controlled with greater prediction and precision than by previous methods. And the second, the introduction of genes into unrelated species is not possible to achieve by traditional methods. The application of genetic engineering is to food production is intended to enhance the useful and desirable characteristics of the organism and to eliminate the undesirable ones. The overall aim of the food industry with respect to GE will be to improve the quantity and to increase the quality and properties of existing food products, to produce new products, and of course, to improve financial returns. Generally, consumers have always shown a willingness to pay more, better, and more convenient products and to reject products that do not achieve their expectations. New agricultural biotechnology now offers a major opportunity to take food products to public demand. Many benefits that GE offers in agriculture include, I'll tell you some of the benefits. First, disease and pest resistance, weed control, animal growth hormones, improve food microorganisms, novel products, improve keeping qualities, tailored products with improved qualities, etc. These are the, you can say, benefits. It is then a matter of choice for the consumer to consider between products of traditional agriculture or agricultural biotechnology based on their needs, targets, and aspirations. Modern agricultural biotechnology can also be used to improve orphan commodities. These are crops which receive little international research attention and which are very important to developing countries' agriculture. Some example of these commodities are cassava, sweet potatoes, yams, and legumes such as cowpea and bean. Conversely, while agricultural biotechnology presents enormous potential for healthcare and the production, processing, and quality of food by genetic manipulation of crops, fertilizers, pesticides, vaccines, and various animal and fish species, the implications of these new biotechnological processes go well beyond the technical benefits offered. Associated with genetic manipulation are diverse questions of safety, ethics, and welfare. The risks from any new technology are divided into those inherent to the technology and those that transcend it. Technology inherent risks of agricultural biotechnology, such as undesirable mutations, must be dealt with appropriately by the biological sciences. The transcendent risks are outside of the biotechnology. As you can see, they are the social, economic, and cultural factors that may distort or obstruct its benefits. The major negative impacts of the agricultural biotechnology include, next slide, please. Okay, you are already there in the next. Safety problem. First is very important, safety problem. In the 1970s, when genetic engineering experiments with microorganisms were first being developed, many molecular biologists believed that the process was unsafe and that manipulated microorganisms should be strictly contained and prevented from release to the environment. The fundamental fear was, and with many still is, that genetically engineered microorganisms could escape from the laboratory into the environment with unpredictable and perhaps catastrophic consequences. It was believed that such related microorganisms could upset the balance of nature, or that foreign DNA in the new microorganisms could alter its metabolic activity in unpredictable and undesirable fashion. It is also feared that various technologies being built into the new agriculture biotechnology products will in future make seeds unviable. The undesirable spread of genetically modified species and strains and reduction in the genetic base of the major trade and food crops and animals increase the risk of diseases and encourage genetic erosion. The safety issues has led to some countries or regions and corporate uh, bodies taking steps aimed at banning or suspending the use of genetically modified products. The public has readily accepted medical products produced from GMOs. They are much less willing to accept such procedures with food. Genetic engineering is seen as unnatural and unnecessary in food production. The safety of the human food supply is based on the concept that there should be a reasonable certainty that no harm will result from its consumption. Foods or food ingredients derived from GMOs must be considered to be as safe or safer than their traditional counterparts before they can be recommended as safe. While the original anxieties about genetically modified products were perceived mainly on safety issues, other social, economic, moral, and ethical issues have come more to the forefront. Next, important is sustainability effect. Biotechnology may aggravate the prosperity gap between the developed and developing nations by replacing tropical agricultural exports with products of the developed societies. Increase milk production from fewer cows by the injection of genetically engineered hormones will certainly result in many small farmers being put out of business. The failure of the general agreement on the trade and tariffs, which is GATT, negotiations to reach agreement on reducing agricultural product protection in the developed countries creates an additional incentive to produce substitutes. Next impact is widening income gap, which is inequality. The use of agriculture biotechnology can no doubt provide real incentives to agriculture development, but in socially and politically defective setting, it is more likely to favor a small and powerful minority than to improve conditions for the poor. Without social reforms that enable the middle and lower strata of society to share in the game, such as land reform and special support programs for small farmers, then technological innovations can work against the development of the development goal of equity. Next impact is exploitation of rich indigenous resources of developing societies. Some people fear that multinational firms or even government research institutes could gain control of genes of plants and animals native to the developing societies. It is imperative to keep open access to the genetic riches of the developing world, and at the same time enable the people who have helped to build and preserve this wealth to benefit equitably from the commercial returns on gene exports. Provision should be made in biotechnology research and development to conserve the indigenous genetic resources. Next is less attention to research and development of interest to developing societies. There is concern that crops of importance of developing nations are not being developed. Biotechnology firms who represent more than half of the total investment in research admit they are seeking a profit. Staple crops such as cassava, yam, then plantain, millets, rice, wheat, mustard, rapeseed, cabbage, pigeon pea, etc. are neglected or likely to receive too little attention without continued strong support for public sector research. Next impact is ethical issues of agricultural biotechnology. Ethical concern include a transfer of human genes to food animals and use of organisms containing human genes as animal feed. Products from transgenic organisms containing copy genes that are, ethically unacceptable to some groups of the population subject to dietary restrictions or their religion should be so labeled to ensure choice. Impact of agricultural biotechnology on different countries, you can say high food importers with strong technological potential could benefit the most. Next slide, please. Now, the recommendations on the appropriate use and application of agricultural biotechnology in developing societies. How can we ensure that most of the world's poor and vulnerable populations benefit from the benefits while being cautious of the risks? Individual countries need to identify their specific national priorities and preferences in food production and harness the growing body of science and innovations in genetic engineering to address specific issues. So, first recommendation is adequate regulatory measures. Regulatory agencies and structures can act as gatekeepers for the development and availability of new agricultural biotechnology products. Developing societies will need to develop and implement regulatory measures to manage any environmental, economic, health, and social risk associated with genetic engineering. World Trade Organization Agreement on Sanitary and Physiosanitary Measures, WTO, requires that measures regulating imports be based on sufficient scientific evidence that and that countries operate regulatory approval procedures without delay. Next is public debate. Public debate is essential for new agricultural biotechnology products and processes to grow. People influence decision making by governments through the ballot box or public opinions. Processes that will legitimately bring the voices of the public to the fore are required. If genetic engineering is to improve food production in developing societies, it should be guided to evolve with local, social, and economic production systems. Next, human resource development and training. Next recommendation is human resource development and training. The enhancement of capacities to engage in the analysis and making of policies on agricultural biotechnology should be treated as a priority by national and international programs. Such training could be offered through international agencies such as the International Center for Genetic Engineering and Biotechnology and by the biotechnology industry. Mobilizing the basic scientific skills usually found in universities to solve agricultural problems will require new policy and institutional arrangements and more financial resources. Very important. Next recommendation is public-private sector collaboration. In most developing societies, public sector investment is still the main source of finance for biotechnology research and development. For public research institutions in Africa, in India, in Malaysia, Nigeria, to access scientific information and investment in genetic engineering, they will need to create strategic links with private companies in industrialized countries, and there is a need for greater public-private sector collaboration in relation to agricultural biotechnology and its applications to problems in the developing world. Next recommendation is intellectual property management. One of the major issues that will affect the application application of biotechnology in agriculture is the protection, is the very important, is the protection of intellectual property. The lack of patent protection is major disincentive for private sector investment in biotechnology in developing societies. Admirers claim that intellectual property rights, IPRs, will encourage innovations and enable innovators to reap a just reward for their investment in research and development. And the last recommendation is support from international development organizations. There should be increased support by international agencies for the development of agriculture biotechnology in the developing societies. This could come in form of loans to undertake specific biotechnology research projects in agriculture, new joint ventures to help local companies participate in commercial development, etc. So such organizations include the World Bank, international finance corporations, rich nations like US, private biotechnology companies, and non-governmental organizations like NGOs. So, next slide, please. With this, I conclude my talk. And once again, I would like to congratulate and thanks to Department of Biosciences, Christian Eminent College, Academy of Management, Professional Education and Research, Indore, Madhya Pradesh, India, then Research Foundation of India and RFI-CARE, Dr. Jain sir, for inviting me and giving me opportunity to express my thoughts on such a wonderful topic. Very well organized. I would like to congratulate the organizing committee also, and thank you so much. Thank you all. Thank you so much for all the listeners for patiently listening my very exhaustive, I can say, talk. Thank you, one and all present here. Thank you.
Thank you, sir. Thank you very much.
Thanks a lot, sir, for your valuable presentation on challenges and impacts of agricultural biotechnology on developing societies. It will definitely be beneficial for us.
Thank you
once again. Now, today, we are proud to have with us eminent personality, academician, respected Dr. Dragan Skopia, educator, speaker, lecturer, inventor, scientist, researcher, social worker, author, phytotherapeutist, phytonutritionist, agroecologist, natural health coach, Skopje, Macedonia. Welcome, sir. Are you present here? Dragan, sir? Thank you very much, sir, for your words. Now it's my honor to introduce today's international guest and speaker. We are indeed privileged to have a global educator, science, technology, engineering, and mathematics STEM instructor, and an ICT teacher trainer in Greece, respected Rania Lampuman. Currently, she is a STEM instructor at a Greek astronomy and space company, and she is also working at the Greek Ministry of Education at the Directorates of Educational Technology and Innovation, where she writes STEM projects for Greek schools. She is the founder and the international coordinator of many innovative international projects that focus on the United Nations Sustainable Development Goals described in the 2030 Agenda. She is also an author of scientific books for kids. She has received many humanitarian and Peace Award from International Humanitarian Organization. She is a member of several international national committees, professional organizations, and other bodies. She has presented her research in numerous international conferences and conferences, and she has published work in various journals. Ma'am has been awarded many national and international prizes, and she is a Global Teacher Award 2020 winner and a Global Teacher Prize finalist 2019. She has been selected as Global Icon 2020, featured on Fashion Vista magazine among the top 10 women entrepreneurs featured in Fortune and among the 100 most successful men in business. Very warm welcome, Rania ma'am. I don't take much time and invite you to share your knowledge with us.
Greetings from Greece. Very, very honored, very happy to be invited in this scientifically deep conference, and I'm very, very excited to connect with all these amazing scientists today. Thank you. I would like to thank Dr. Dajay for his kind invitation. And today I will try to present multiple perspectives of applied biotechnology in agriculture, since the application of biological sciences in agriculture has become increasingly prominent in the past decade. And of course, the science of biotechnology for agriculture is in its infancy, but it's also already a clear influence. At the same time, it's important to focus on the social concerns and risks, and that's why there is a need of what we call value-oriented research. Why? Because when new technologies are involved in large-scale social changes, public debate and opinion formation on value-based issues can be facilitated by research that articulates and analyzes ethical, legal, economic, and cultural issues. So may I share now my presentation? My computer has stuck. Wait. Okay. Is it visible now?
Yes, ma'am. It is visible.
Yes,
Okay. Can you see also the change of slides?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. So Homo sapiens has altered Earth environment since its emergence. and environmental perturbations caused by humankind have evolved continuously since the beginning of civilization some 20,000 years ago. But in the past century or two, we have changed ecosystems with such intensity, on such a scale, and such speed that a new geological era, what we call Anthropocene, has been proposed. So the impact of mankind in Earth ecosystem is destabilizing the whole period of the past 10 to 12 millennia. It's the period which is called Holocene, which is the only state of the planet that we know for sure can support contemporary human societies. So this is new geological period, which is not yet officially recognized, but all scientists say that we are already reached this point. And as the global population expands to 10 billion people, the pace of change is faster than ever before. There is an urgent need of a paradigm shift to maintain Earth in a safely operating space for humanity and for the millions of species with which we can share this home. So the dramatic negative impact that the massive scale of, for instance, deforestation has caused not only to the habitat of for million species, but also in overshooting key Earth system parameters. This is very important, and also how undeniable deforestation, intensive agriculture with traditional crops are all these major human risk factors pushing the Earth system beyond the bounds of safety. And although our inventiveness is the driver of the problems we face, it's at the same time the source of innovative solutions. The United Nations 2030 Agenda for Sustainable Development represents an important framework for tackling challenges and acknowledges that sustainable development goals cannot progress without strong engagement by science. At the same time, the robust knowledge creation of the recent decades shows that science is accelerating its pace to bring the solutions that society is asking for. The transformative steps which are needed to shift the world onto a sustainable and resilient path need not to leave anyone behind. So the engagement by science, including economic and social sciences in SDGs, must involve all countries, developing alike. And agriculture plays a crucial role in SDGs because it is a main human activity that permits most of the SDGs, from hunger and malnutrition to poverty alleviation, education, gender equality, water use, energy use, sustainable consumption and production, climate change, ecosystem management. So agriculture actually acts as an engine of overall economic growth and development in many economies, notably in least developed countries, where the majority of the poor are rural people. So global agriculture has been successful in providing sufficient food for expanding populations and their changing consumption patterns over recent decades. Per capita agricultural productivity has outpaced population growth. Intensification rather than the spread of agricultural land has been the prime driver of global agricultural productivity since the mid-20th century. And agricultural identification was the only way forward, with the scientific knowledge then available to cope with the dramatic rise in population in recent centuries. So between 2900 and 2000, the increase in world population was three times greater than during the entire previous history of humanity. We're talking about an increase from 1.5 billion to 6 billion in just 100 years. But this agricultural intensification has been accomplished at great expense to the environment, causing water scarcity, soil degradation, ecosystem stress, biodiversity loss, high levels of greenhouse gas emissions, a significant decrease also in forest cover. Livestock is the world's largest user of land resources, and grazing land and cropland dedicated to the protection of animal feed represents almost 80% of all agricultural land. And agricultural food production currently account for about 30% of energy consumption and about 1/3 of greenhouse gases. Agriculture accounts 40% of the Earth's land surface, 70% of the world's fresh water, with predictions that irrigation demands will increase by up to 100% by 2050. So during the 20th century, the area under irrigation, the number of agricultural machines grew about twofold, fertilizer consumption by fourfold, and nitrogen fertilizers by sevenfold. The world consumed 186 million tons of fertilizers in 2016 and about 7 million tons of pesticides in 2017. Global fertilizers and agricultural chemicals manufacturing industry reached a revenue of 377 billion in 2018. Anyway, transformation of obviously of agriculture is likely to be the greatest challenge of the agenda, and debates of the future of agriculture are being framed into two different ways. First of all, what we call organic agriculture and sustainable and intensification of agriculture. So organic farming relies on the use of natural inputs and ecological processes to make farms more sustainable, and deliberately excludes the use of external chemical inputs such as fertilizers and synthetic pesticides, as well as GM crops. Organic agriculture practices are followed by 204 million farmers in about 87 countries that observe some sort of regulation certification, and accounts to only 1% of the global agricultural land. On the other hand, we have a second category called sustainable intensification, which takes the idea of both conventional agriculture and organic farming, and emphasizes the use of locally adapted management systems and gives preference to in-farm inputs. So the potential for organic culture to feed the world is very debatable. And comparisons of yield data between organic and conventional agriculture report a lower yield for organic agriculture from 8% to 50%. So some studies suggest that adoption of organic agriculture under conditions of optimal performance might close the gap between organic and conventional crops, but these findings have been fiercely refuted by numerous scientists who claim faulty methodology. There are also experts who say that we talk about this issue of supply of nitrogen. In organic fields, nitrogen they say that it must be supplied into biological and fixation of intercrop rotated legumes by the ex situ biological and fixation through use of organic material that could be crop residues or grain manures or biofertilizers. So advocates of organic fertilizer often claim that there is enough organic material to supply the amounts as per the crop demand for high yield, but the facts are different because organic materials are not universally available in large quantities. When we estimate organic productivity, organic farming supporters do not properly acknowledge that must be allocated and to supply nitrogen for the growth of non-legume crops. And the data available clearly suggests that extra land and water would be needed if such sources of nitrogen were to be promoted. So by using more land to produce the same yield, current organic practices may ultimately accrue larger environmental costs. And the strategy of sustainable intensification is actually to get more from less. It seeks to produce biomass needed by the growing human population on less land with less adverse impact on the environment. And this does not specify particular technologies or practices. In particular, it focuses on increasing yields of farmland as a way to spare forest and other cultivated land. A recent study of the effect of land sparing that includes 17 organizations around the world concluded that more intensive agriculture that uses less land can be a way forward. And this study found that organic nitrogen improved yields inorganic nitrogen improved yields without increasing greenhouse gas. Intensification also produces fewer pollutants, causes less soil loss, and consumes less water. This sustainable intensification acknowledges that there is no perfect solution due to the multi-objective nature of sustainability. The process, of course, is dynamic here. Technologies and practices will not fit everywhere ever. For instance, in Africa, population growth is shifting to Africa. It's projected to equal the Asian population in 2000 100. Africa, for instance, needs new sustainable intensification approaches to deal with the challenge of feeding an exponentially growing population, and the solution will not come only from new management strategies. It is clear that the use of proper crop varieties is the ultimate solution here. So improve the crop varieties suited to local conditions and weather extremes, as well as pest and disease-resistant cultivars, increase yields and reduce pesticide use. We talk about plant biotechnology, which is important. Why? Because it has the tools to tailor crop varieties to the environment. They help to meet the goal of producing more food without degrading ecosystems. So under the umbrella of sustainable intensification management systems are developed to build on farm soil fertility and in situ nutrient application, sharing cropland with complementary species that do not compete for resources. This has been praised as a win-win management approach. For instance, trees are ideal partners for crops because they do not compete for the same source of nutrients, and tree roots go deeper into the soil and get nutrients and water from sources that are unavailable to crop or species. So well-designed agroforestry systems for sustainable intensification represent an interesting sustainable strategy, particularly in those areas where the need for landscape restoration is associated with the need for increased food and biomass production. And here we can see why agroforestry is important, because it does not require extra land, because trees are planted around and among crops and pastures. And also, depending upon which wood species are used and how they're managed, agroforestry can build up on farm soil fertility, increasing resource use efficiency, at the same time reducing nutrition runoff. Recent Africa has demonstrated that the integration of fertilized trees and shrubs into conventional agriculture can dramatically enhance soil fertility and food production. And other also benefits of agroforestry include more favorable microclimates with enhanced biodiversity and reduced wind velocity, enhanced suppression of insect pests and weeds, decreased levels of soil erosion, increased water infiltration, improve production potential by increasing crop yield, and diversification of production by generating products for the in the crop trees. So agroforestry is associated with farming in tropical and subtropical arid regions. However, there are also opportunities for agroforestry in temperate regions. So the challenge here lies in the political will to promote the research required to the adoption of agroforestry as a mainstream agricultural management approach, and a number of plant biotechnology innovations are ready to boost agroforestry systems, including trees with a variety of GM traits, which is a very controversial issue, however. So since we're talking about agricultural biotechnology, most speakers have already explained what is a range of tools, including traditional breeding techniques that alter living organisms, parts of organisms, modify products, improve plants or animals, develop microorganisms for specific agricultural use. Here's some types of these: genetically modified crops, developing biofuels, improving plant growth, improving plant seed quality, improve animal health and breeding, learn more at the fruit grower supply. And what is the purpose? Of course, the purpose is to address problems in all areas of agricultural production processes, and this includes also the plant breeding to raise and stabilize health, to improve resistance to pest diseases and abiotic stresses such as drought and cold, to enhance the nutritional content of foods. Little brief, I just want to mention that one of the first applications of biotechnology was in agriculture. For centuries, farmers have been manipulating the genetics of plants and animals in order to obtain certain traits or enhance them through a process known as selective breeding. And a very early example of conventional agricultural biotechnology would be how farmers kept on cross-breeding different varieties of wheat until the much-needed disease-resistant trait was obtained from the resulting new variety of wheat. Recently, as the science evolved, by 1970, scientists learned to make changes to DNA, so at the molecular level to transform them into genetically modified organisms. This is called genetic engineering, and this involves using scientific techniques to modify the genetic material of plants and animals, microorganisms. And later, we were able to isolate specific traits from plants, such as increased yield, pest and drought resistance, even pesticide or herbicide resistance. By the end of 20th century, the technology moved further. We have the first genetically modified food in 1994, the flavor saver tomato. And in 2019, we have reached the level now where we can not only change, but also transfer specific genes from one organism to another. An organism developed through such genetic engineering methods is known as genetically modified organism or transgenic organism. And these are basically foods that come from transgenic plants and are called multiple names such as genetically modified products or biotechnology enhanced foods or biotech foods or Franken fruits or genetically modified GMO. I need also to make a difference between traditional biotechnology, genetic engineering. This is what we said before, how conventional breeding involves processing of breeds in a controlled manner. And this way, the breeder could certainly control which two breeds to cross, but what goes on at the genetic level is out of his control. And there, in traditional biotechnology, all the traits are mixed and randomly end up in the offspring, which means with the desired trait, there could be a few unwanted traits. For instance, like a plant could have a high crop yield, but at the same time, very low pest resistance, which could be deadly. So all these methods take up quite a long of time and effort to work. A great chunk of this is used up in obtaining desirable traits and removing undesirable traits from the genes. And on the other hand, we have genetic engineering, which allow us to segment the section of DNA that codes for the gene of the desired trait and transfer it to the DNA of a new transgenic organism. In the same manner, we can remove undesirable traits from the gene sequence of an organism as well. And this use of recombinant DNA technology allows us to achieve changes much quicker than with traditional breeding techniques. Research suggests that 60–70% of food products may derive in full or partially from crops developed using new techniques. And in general, farmers have embraced this change to newer methods so well that 1/3 of the corn as well as 1/4 of soya bean and cotton grown in the USA is now sourced through genetically modified crops. Here's a transgenic crops that have been approved. Corn, potato, this the case of cotton is interesting to see that the highest production is of cotton, but technology, corn and soya beans are resistant to glyphosate. Here also we needed to see that glyphosate is a herbicide that kills almost every plant that gets in its way, but the presence of the glyphosate resistance gene in the soybean DNA allows the farmers to openly spray glyphosate over the farmland without any harm to the crop. So what happened with cotton and corn was that the DNA of a naturally insect-killing organism, Bacillus thuringiensis, was introduced into their DNA, and this organism kills also most of the deadly pests. We're talking about here a new revolution of pest control for the farmers. Of course, when we talk about all these products, we talk about at the same time risks and benefits. There are benefits, of course, and why? Because they haven't been scientifically proven until today completely. So, what are the advantages of applying biotechnology in agriculture? First of all, higher crop yield. With the rising population, demand for food has become necessary to increase the yield of farms to fulfill that demand. Biotechnology has made it possible by strengthening disease and drought resistance. And what they do is that they select the specific genes of disease resistance, inject them into DNA of plants to make them resistant. Drought resistance is also quite the need for crops growing in dry climates. Secondly, higher protection of crops. Farmers use new developments in the section of pest control because if not, there is a risk for severe lowering of yields. However, some farmers who insist on growing organic are recommended to and make use of applying the natural insecticide externally. Increased nutritional value because biotechnology does not only make plants be more resistant to worsening climate change, but literally makes them better. Specific isolated genes can never be inserted into the genome of plant species to increase the nutritional value. This theory was put in practice with rice, one of the most eaten foods in the world, and it was discovered that rice contained vitamin A in their genes, but while growing, those genes would not be turned on as to say. So what science did is to reverse the process and thereby activate those genes during growth. This means that the nutritional value of many other food crops could possibly be increased using recombinant DNA technology. And considering the rising food demand, this could be an answer to solve malnutrition. Other examples could be higher protein content in soybeans or potatoes with increased amino acids and starch content. Another advantage concerns enhancement in food production processes. Chymosin is an enzyme produced from genetically engineered bacteria. It was the first food product to get approval for commercial production, and it replaces an active ingredient, the calprenate, used in cheese manufacturing processes. So the genetically engineered enzyme has become so integrated into the process that it's now in 60% of all cheese manufactured worldwide. And why? Because of the higher purity, continuous supply, and up to a 50% cost reduction. Better flavors, believe it or not, genetic engineering can also alter the taste of food products. This is done by promoting the activity of enzymes that work to transform aroma precursors into flavor compounds. And fresher produce, increase shelf life. Agricultural biotechnology can increase the shelf life of GM food products, which could help in reducing waste as well as to allow consumers access to fresher produce. So by allowing the food products to stay ripened and prevent them from over-ripening, minimal food will go to waste. And what are other benefits for environment? We said before how transgenic crops became inherently resistant to pesticides, and this led to much reduced use of pesticides. Only the organic farmers now have the need of applying the natural insecticides externally, which is quite a win. So with reduced use of pesticides, there is reduced pesticide residues of food products. Improvements in developing countries, this can save people from malnutrition. And however, there is always a dark side of new inventions, because what research says that there people get quite confused in statements regarding biotechnology projects. They suffer great anxiety, which was possibly derived from a lot of misinformation, in many cases inadequate information. And also environmentalists, policymakers, and legislators believe that we haven't made sufficient effort to understand the risks associated with mixing biotechnology and agriculture. And this, that it will have long-term impacts as well, especially when one mistake could affect generations to ask. So it's something that these concerns are understandable, and the research must be go forward. So what are the disadvantages? Usually allergens and toxins. Most of us know that what an allergic reaction is. About 20% of the total population in the world is allergic to one or two food products. So a major concern is the risk of using new allergens into foods that were initially considered safe and free of all allergens. Antibiotic resistance. When plants are infused with new desirable traits, the antibiotic resistance genes are used to find and trace the traits. So this technique tells us that the gene transfer was successful. However, since this technique uses these antibiotic resistant genes, there is a concern that new antibiotic resistant bacteria could emerge, which will be difficult to tackle with conventional antibiotics. And what called potential super weeds, transgenic plants, it's a similar concern that the transgenic plants could pollinate with unwanted plants and thereby relay the gene of herbicide resistance or pesticide resistance into them, thereby converting them into what we call super weeds. Gene escape. Some believe that the genes could get into the weeds and then those plants could escape into the wild and lead to damaging changes of ecosystems. But things is that the biotech crops have limited growth and seed dispersal habits, which doesn't allow much of a chance to pollinate. Other than that, these plants need constant attention by humans for them to grow properly. And effect of non-target species, many environmentalists believe that once those transgenic plants are released into the environment, there is a lot of uncertainty of what could happen. Although the transgenic crops are tested more times than one count, we can't foresee what could happen. In pesticide resistance, we know that a valid concern is that insect pests could develop resistance, but they could bypass the crop protection features of those plants and be quite damaging. Loss of biodiversity in organisms, the extensive use of agri-tech varieties of seeds have made some agriculturists fearful that this may hurt the biodiversity of plant species. Food labels, people argue that they have a right to know what they are consuming, that this is why they request that special labels should be put on food products derived from genetically engineered crops. And suicide seeds, this is also known as genetic use restriction technology. Farmers are forced to buy new hybrid seeds every season because the second generation seeds from biotech crops are always infertile. And this infertility is due to terminator genes present inside GM seed. It's like a single-use product. So in conventional family farms, farmers are used to store the seed to plant in the next season, but that cannot happen if you would like to plant transgenic crops. This means that to reap the benefits of plant biotechnology, the farmers will have to enter the economic cycle of major biotech seed companies. So we'll talk about this, the pros and cons. Here are the safety regulation by technology food projects by United States Department of Agriculture, USA Food and Drug Administration, environmental and some regulators. And another interesting issue that we can mention here is what we call phytoremediation, because in the future, transgenic plants may be grown to remove or detoxify pollutants from soil already to convert the highly toxic organic mercury in soil into less toxic elementary mercury, which is volatile. And another approach is to accumulate mercury in plant tissues where it can be harvested, extracted, or disposed for more safety. So these phytoextraction plants are already capable of accumulating more than 1% of their biomass as mercury, and crop improvement techniques, including genetic engineering, may be able to increase that fraction. The environmental impacts of these mitigation strategies are probably, of course, scale dependent. I mean that while phytoremediation by volatilization would have local benefits, the large-scale consequences are less clear. If the total amount of volatile mercury created is relatively small, it will be probably of little environmental consequence. And therefore, small-scale use of the volatilization strategy could be beneficial for the environment. If it's large, then the levels of atmospheric mercury may rise. And we don't know about what happens now about the destructor that there are that all this has led to segmentation of markets for genetically modified GM-free crops. United States, the creation, for instance, of marketing standards for certified organic crops that do not contain transgenes has established a market niche for producers. On the other hand, at the same time, the existence of a market for non-transgenic crops creates a new kind of environmental risk from transgenics. And such changes may have already both economic and environmental impacts. For instance, widespread use of herbicide-tolerant crops has changed patterns of herbicide use, resulting in new types of damage claims from herbicide drift, forcing farmers to adjust their farming practices accordingly. And this way, farmers may incur legal liability for technology fees as a consequence of neighboring fields inadvertently pollinating a crop leading to transgenic seed production. And we all know the case of combinations of transgenic traits. In the future, there's expected to be proliferation of novel transgenic traits, engineered crop cultivars involving multiple genes. Already, transgenic crops expressing multiple traits have been commercialized, such as cotton, wheat, and corn, that both have pesticidal properties and herbicide tolerance. Also, Many cultivars with stacked genes with multiple transgenic traits are expected to be commercialized soon. In the future, there's expected to be this proliferation, and also we talk about another issue is the monopolistic control of seeds and other products. And the access to information is also important because information explosion in the field of technology is being assimilated in various databases, but the information is not always available freely due to restrictions imposed by patent regimes. And the free access to information at all levels is fundamental to the rapid improvement of livestock, forestry, and fish species. This is particularly important for developing countries, which are not in position to create new technologies. Partnership between seed-rich and gene-rich molecular biology and genetical engineering research call for high-level investment of financial and human resources. So most developing countries are able to commit the needed financial support. So the costly biotechniques and products are therefore out of reach of the majority of poor institutions and people in the developing world, which is the contradiction here. And the private sector research in biotech development has therefore tended to be confined to well-endowed countries, communities, and commodities. So to meet these challenges, the public sector will need to more advanced technologies, which are often being developed by the private sector and are proprietary, and the marginal areas inhabited primarily by farmers and other people who are not poor in everything. Genetic resources evolved through ages and local knowledge accumulated and enriched over generations in such difficult habitats are unique and invaluable. So a good part of genetic resourcing, the associated information and knowledge is conserved in public sector, national and international organizations. GM, herbicide tolerance traits, also something that contributes to substantial contribution to sustainable agriculture, allows the application of more environmentally friendly herbicide in a more rational way, enables the adoption of conservation tillage. And we all know about bio fertilizers that are composed of microorganisms such as algae and fungi, which improve the overall fertility of the soil. Although the effect of these biofertilizers is not as immediate as that of chemical fertilizers, they can provide nutrients contain the soil to crops more efficiently in the long term. However, because soil conditions farmers vary from region to region, from crop to crop, the types of microorganisms that can be applied as vary as well. So the difficulty in handling also microorganisms poses a storage issue. We talk about smart agriculture and agricultural biologicals. While agricultural biologicals expect to be means to achieve sustainable agriculture, they are still in research development stage. However, trying to shift agriculture with less environmental impact is strong, and policy support is considered for finding solutions. So we have attempts to utilize smart agriculture and data obtained using analytical approaches such as omics analysis and phenomics technology. Smart agriculture optimizes agricultural operations based on collected data and yield information, including sunshine, rainfall, temperature, and humidity in the field. Omics analysis is comprehensive for biomolecules in crops, microorganisms in soil, and phenomics technology enables the analysis of the shape and growth status of plants. So if the development and optimal use of these technologies are promoted, agricultural biologicals may become alternatives or may be superior to pesticides and chemical fertilizers. It's interesting to also highlight a few issues about food systems so far, because it's challenging of delivering the food sustainable way goes beyond agriculture. First, we can mention all these consumption behaviors. Reducing the overconsumption of calorie-dense food will improve the overall sustainability of agricultural food systems, while addressing a major threat to health, because this concerns the case of obesity of many people, and the case very important of what we call indigenous crops, because amelioration of crops at essential parts of the diet of those with poorer regions can have a major impact on food security. So crops indigenous to these regions have the potential to mitigate the impact of climate change and food protection, because they tolerate fluctuations in growing conditions and are resistant to local diseases and pests. and compared to the world major crops, indigenous crops are better adapted to the niche environments. Another very important issue is what we call food waste. And we see how roughly 1/3 of the food in the world produced for human consumption every year gets lots of wasted. And this we need to minimization of losses could indeed be a great help in achieving global food security. So a number of GM traits have been developed that improve food storage stability in crops. This is very, very important. Apart from the food waste, we have the uneaten food accounts which account for uneaten food accounts for 8% of the greenhouse gas emissions. The pattern of food waste is different, of course, between high income regions. In high income countries, food waste is higher. For instance, developed regions such as European Union are more concerned in reducing food waste at an extended post-harvest stages. All these issues are very important. We need new strategies to solve these problems. Are what we call the low-hanging fruit, and are prioritized in European countries and public-private sectors cooperating to bring practices, protocols, and cold chain equipment. We mentioned before how low-income regions suffer more from losses at stages of harvest and strict post-harvest. And plant biotechnology innovations could bring solutions to reduce losses during harvest and post-harvest, are being overlooked usually. Why? Because they are still a sensitive subject with no political clarity. We talk about the role of science here. The major forces that shape our modern world are science, economy, and society. And these forces are interdependent. Science has provided, of course, many tools to help humanity to reduce global environmental risks and promote global sustainability. But science alone not help solve problems and shape the future. So these three pillars that sustain human civilization, society, science, and economy must be in correct symmetry and based upon solid ethical and moral grounds. There is no place for nationalistic, xenophobic, racist, or anti-science rhetoric. Why do I say that? Because, people's attitudes towards plant biotechnology is usually influenced by misinformation, by the bad information, or lack of research, and massive campaigns against transgenic crops, etc. So, before concluding, we can see what are the concerns about the risks and contradictions. Of course, we're talking about the depletion of biodiversity and poor access to tailored genetic resources. This is a contradiction. The adverse environmental effect, negative effects on human health, and the technology transcending concern of widening the inequity of poor access to the new emerging technologies and products on part of developing countries and research poor people, the majority of small farmers, is another major contradiction. Any attempt to mitigate environmental risk must be mindful of the fact that avoiding one risk may inadvertently cause another greater risk. And this might occur if attempts to avoid risks from commercialization of transgenic crops resulted in the adoption or continuation of commercial agricultural practices with greater negative environmental impact. If, for example, a lower yielding non-transgenic crop is chosen instead of a higher yielding transgenic alternative, the result could be that fragile lands would need to be planted in order to produce needed amounts of food. And also, unwanted environmental outcomes could also occur if regulatory oversight itself has unintended consequences. The largest, also a most complex factor in making cooperative judgments of agricultural technology is that the main goals of technology are to provide more and better food for public consumption, to reduce the cost of production. But the circumstance where food supplies and adequate people may become more willing to accept perceived trade-offs between environmental quality and food supply. And also changes in food production that lower prices are especially beneficial to the poor, because even in times of plenty, poor people spend a proportionally larger share of their income of food. So the context this context must take account of the need-based argument for expanding capacity and production. Both the human and environmental dimensions of the need for increased food production are critical to a balanced evaluation of the risks. And here we can see what is the need for improved scientifically sound protocols to detect effects of transgene products or non-target organisms, a need for improving understanding of the environmental effects of transgene movement. So we need to develop a theoretical and empirical understanding of the kinds of environmental effects. And also there is a need for research on the effectiveness and efficiency of present regulatory systems to develop approaches to enable regulatory systems to improve scientific principles in data. Research is needed to evaluate environmental risks associated with conventionally produced cropland varieties, and also useful to fund research, to identify and investigate possible environmental hazards of many novel transgenic organisms. Of course, we're talking about future policies, a huge discussion. The policy questions will be raised in the future by new transgenic crops and many other factors that influence shifts in policy perspectives will differ from those that are applied today. Discussion new kinds of transgenic crops, environmental risks, focus on policy discussion, research needs to address future. And for concluding, I would say that, of course, it's a necessary evil. Agriculture by technology is a necessary evil because we have to bear it, to tolerate it if we want to rise the food demand. Analyzing population graphs, the food price is inevitable by 2050 because there will be a 70% higher demand of food. So the only way we can fulfill this huge demand is by using such applications by technology to improve crops, increase food supplies. A development of plants is also necessary as we'll be encountering worse weather conditions, climate change worsens. And also, of course, given all these pros and cons that we have far mentioned, it's difficult to make a fair decision if we should embrace the use of biotechnology in agriculture. It's a debatable, questionable issue. But what is firmly believed is genetically modified crops could have some unprecedented results on human health and environment. This is why companies that make genetic crops should extensively test these products before introducing them into the markets. Thanks very much for your attention. And
Thank you, ma'am.
Thanks a lot, ma'am. We sincerely appreciate you for taking out time from your busy schedule for providing us with such valuable information. Thanks a lot once again.
thank you very much.
Now it's a time to express our gratitude to all. And for this, I would like to call Neha Joshi, madam, to express a vote of thanks for inaugural ceremony. Over to
Thank
Neha,
you, ma'am.
ma'am.
Thank you, ma'am. Is my voice audible, ma'am?
Yes, ma'am.
Yes, ma'am.
Good afternoon, everyone. It has been great pleasure for all of us for successful accomplishment of inaugural session of this international conference entitled as Challenges and Prospects of Biotechnology, Environment and Agriculture Researches, organized by Department of Bioscience, Christian Avenant College, Indore, in association with Research Foundation of India, RFI-CARE. First of all, I express my gratitude to all our respected international and national guests, respected Dr. S.K. Mishra sir, respected Dr. Supriya madam, respected Dr. Sudhakar Umle sir, respected Professor Dr. Pratik Rajan sir, respected Dr. Dragon sir, respected Ms. Rania madam, for accepting our invitation and delighting today's inaugural session with their informative knowledge. I'm thankful to Research Foundation of India for this wonderful collaboration. I'm also thankful to JHERF World Virtual Conference Forum, World Federation of Science and Technology, International Research Guide Federation, Academic Research Guide Association, and EDU Academic INC for their sponsorship. I'm much obliged to our patrons, respected Dr. Saurabh Jain sir, Chairman and CEO RFI, respected Mr. Sameer Mir sir, Chairman Christian Avenant College, Indore, respected Professor Y.S. Chauhan sir, Director Admin Christian Avenant College, Indore, respected Professor Dr. Ashok Kumar Gupta sir, Chief Managing Director RFI, respected Dr. Rajesh Vyas sir, Principal, Christian Eminent College, Indore, respected Dr. Ajay Jain sir, National Coordinator, Research Foundation of India, respected Dr. Sanjay Prasad sir, the State President, Madhya Pradesh RFI, respected Dr. Manish Dubey sir, President, Central India Board RFI, and respected Professor Rajesh Shah sir, Vice Principal, Christian Eminent College, for their continuous support and guidance. My special thanks to attendees of this inaugural session. Thank you so much, everyone, for making this inaugural session successful. Now, it is time to move ahead towards our first technical session. And I would like to take permission from Dr. Shivam Pratap sir, Head of the Department of Bioscience, Christian Eminent Colleges. Sir, shall we start our technical session?
Yes, ma'am. Move ahead.
I also want to take permission from Dr. Azmat Alam sir and Dr. Anubha Pandya ma'am. Sir, can we start, ma'am?
Yes, please go ahead. Please go ahead.
Yes, sir. Thank you so much. Once again, welcome all dignitaries in first technical session of International Conference on Challenges and Prospects of Biotechnology, Environment and Agriculture Researches, organized by Department of Bioscience, Christian Eminent College, Indore, in association with Research Foundation of India, and sponsored by JHERF World Virtual Conference for World Federation of Science and Technology, International Research Guide Federation, Academic Research Guide Association, and EDU Academic INC. Today, we have with us a very well-known academician and multi-talented personality, respected Dr. Azmat Alam Khan sir, respected Professor sir and Chief Scientist Officer on special duty to Vice Chancellor and Principal Investigator, ICAR Poverty Seed Project, as our guest and speaker in our technical session. It is my proud privilege to say that Dr. Alam sir is associated with many assignments like Associate Director Research, Co-Coordinator DBT, Nodal Officer Action Plan on Rural Leg, Model Officer for Development Work under Prime Minister Development Program FPSCNA Act. Dr. Azmat Alam sir has been bestowed with several honors and awards such as Best Research Paper Award, Best Presentation Award, Best Student Award during PhD, and many more. Sir completed almost more than 10 research projects and funded by renowned funding agencies such as ICER, DBT, DST, BV Rao Poultry Research Foundation, etc. Dr. Alam sir published more than 100 research papers in national and international reputed journals. It is my honor to say that sir is a member of professional societies such as Indian Poultry Science Association, Indian Association for Advancement of Veterinary Research, SKUST Journal of Research, Indian Society of Animal Breeding and Genetics, World Poultry Science Association, and Indian Society of Animal Product and Management. It is a matter of great pleasure and honor that sir is here with us to address us, and we, with immense pleasure, welcome you wholeheartedly. Without taking more time, I request sir, please share your knowledge with us. Respected Dr. Azmat Alam sir. Sir?
Thank you, Dr. Neha, and
Thank you, sir.
thank you, my friend Dr. Shiv Om Pratap. All the dignitaries here, all the learned professors, and all the learned participants of this conference. The greetings and a very good afternoon from my side. I am from Kashmir, which is considered as a very beautiful place on earth. This time we are meeting in an online mode. Again, thanks to the technology which has placed, which has enabled us to be once we are thousands and thousands of kilometers apart, and we are now together because by virtue of this technology, and it is because of this COVID which has taught us so many things. This would have definitely maybe some 10 years after present times, or 20 years afterwards, we would be doing the same thing. But COVID has accelerated our pace. So thanks again to the technology, and thanks again to God. He has, although we may think that this COVID has been sort of a trouble to us, but it has proved to be a blessing, and we have been able to little bit we have been able to accelerate our progress in terms of this online technologies and online sharing of knowledge. Is my presentation visible there?
Yes, sir, it is visible.
Thank you very much. Uh, well, this gives me a great pleasure to present some of my views there in this conference, where I believe the hardcore biotechnologists are there in the conference, but I will be talking of biosciences, and particularly the agriculture and allied sectors, and more precisely of poultry sciences, and we will be discussing some innovative approaches so as to uh, that approaches lead us to sustainable poultry production. So I'll be speaking on some novel circular bioeconomy models for sustainable poultry production. In this, you know that our population is growing at a rapid pace globally, and we have more mouths to feed, but the land is decreasing. Other resources like water and energy resources that are decreasing, but the people are increasing, and we have by 2050 there will be some 10 billion humans living on the earth, and they have to survive on those very limited resources. The resources which we have, they are limited, and these resources are to be shared by the growing population. Similarly, in terms of land, if we see in 1970, we were some 4 billion people living on this earth, and per capita land sharing was 0.38 hectares. In 2000, we grew to 6 billion people, and the per capita availability of land was 0.23 hectare per person. And in 2050, we will be 9 billion people with per capita availability of land as 0.15 hectare. So the resources available per person, they are decreasing. On the other side, the demand is increasing. Here I have some data. When we compare the demand in 2005 with the demand in 2050, suppose we talk of mutton, it was 13 million. Now it is going to increase 25 million. Similarly, pork, almost you can see from this graph, the demand for all the things, it is almost doubling. So in this scenario, we have to look for the solutions, and one of the great solutions is coming from our bioeconomy. But again, when we talk of bioeconomy, we have to talk of sustainability. What is sustainability? Sustainability means that it is that development that not only meets the need of present, but we should be able to project the future needs. We need by fulfilling the present needs, we should not be compromising our ability to meet the future needs. So we have to, there are three important E's. E standing for environment, E standing for economy, and E standing for equality. So this is, or equity, what we call, that means equal distribution of the resources. Everybody should have equal access to the resources, and we have to take care of our environment, and of course the economy. So we have to use our resources judiciously so as to support the future generations and therefore ensure sustainability. So here comes the role of circular bioeconomy. If in this diagram, if you see in the linear economy, we have raw materials, we then it goes into the production chain, we use it, and then we generate waste. But in the circular economy, there is an element of recycling. We have such designs; those are sustainable. We go for production and then remanufacturing of the things. We talk of not only use but reuse also, and we talk of waste collection and putting that waste into reuse. Thus, by moving from linear economy to circular economy, this ensures our sustainability. Now, since I am mostly trained in poultry sciences, I'll be giving some of the examples of how, what sort of bioeconomy models in poultry production we have been using at our university so as to move towards sustainability. We'll be talking about poultry production in a natural way, not in that factory farming mode, but in a natural way. Then we will be talking about integrated farming. Uh, that is, we have a horticulture integrated farming model in place here. Then one more, this nursery raising, which is now emerging as a new rural enterprise. Then we'll be talking something about this easy hatch, a new innovation, which again is a step towards sustainability. Then we'll be talking something about alternate poultry, particularly the waterfowl. I believe in morning session, uh, Professor Mishra, he was also talking about ducks, etc. In that direction, also we have done something for the sustainability. And then lastly, will be because our place is a very cold place. We are facing a chilling winters and very long 4 or 5 months chilling winters. For that, to rear chicken in those chilling months, we are going for this cold conditioning. Here we are, maybe the biotechnologists in the group will try to will be able to understand it better, and they will try to, they will be interested in this approach where we are trying to condition the chicken so that they can grow well under these cold conditions. So first we will move to this poultry production in a natural way. You know, we have in all over the country these poultry development programs have been going on, and we have many programs from ICAR, from state departments of animal husbandry, these poultry departments, and other rural development departments. We have such programs going on where they promote poultry production. So that is the natural way of poultry production. That means backyard poultry. They promote the industrial poultry. It is okay. We have very big, large poultry farms all over, but then they promote in the rural areas or even in the city areas. They promote this backyard poultry farming when every person keeps few number of birds that live on those kitchen wastes and other things, and then they provide some poultry meat and poultry eggs. So if you look on those ongoing backyard poultry development programs that are carried out in the country, they have one peculiar thing that birds which are reared in those on all over the backyard poultry development programs, they are dual purpose birds. Dual purpose birds means they are equally good for meat production, they grow fast, and they are equally good for egg production. Uh, but what we have done, we have tried to do a different kind of intervention in this regard. So if we see the ongoing backyard, what the routine backyard poultry programs that are carried out, those breeds I told you, they are the dual purpose breeds. They grow very fast. Since they grow very fast, they are fat birds. they are not able to escape from the predators. Since they are fat, हिंदी में कहेंगे उनके पीछे कुत्ता या बिल्ली दौड़ती है, उस मुर्गी को पकड़ के खा जाता है. तो ये एक प्रॉब्लम है. और दूसरी बात, when we distribute these birds among the farmers, when they grow very fast, so farmer gets attracted and it uses those birds for table purposes, and they do not last long. They are consumed, their meat is consumed. So egg production के लिए वो रह नहीं पाते हैं. They remain with the farmer for a very short time. But what we have done, we have tried to introduce the true layer type bird in the backyard poultry system. Since these birds are lean, thin, and agile, they are able to escape from the predators, and therefore, farmer also has a very less likelihood of using these birds for the table purposes because they are lean and thin birds. I think Shivam Pratap knows these are those leghorn type of birds. They do not have much meat, so the farmer is likely to keep these birds for the egg production for some long time. I have calculated this investment versus benefits accrued under this project. This was actually our NABARD funded project, and the total fund which NABARD gave us was 7,22,000 rupees. And these now in this lower table, benefits accrued, these are the benefits. What we did, we distributed those birds among the poultry farmers and various beneficiaries. There were around 240 beneficiaries. To every beneficiary, we gave 10 birds. So there were 2,400 birds in the field. There was mortality. When we calculated what was the mortality, it was 7%. But for calculation purpose, to make calculations easy, I have taken it on the higher side. So let 10% birds have died. So 240 hens died, and only 2,160 hens made. And the egg production was from these 2,160 hens. So when we asked farmers how many eggs did these hens lay, they said some farmers reported 220 eggs. But on an average, every farmer, when we drew the average, it was 170 to 180 eggs per hen per year. Again, for calculation purposes, we take the lower side figure of 150 eggs per hen. So we got 3,24,000 eggs. Now the value of the eggs. These farmers, they either consumed those eggs or sold those eggs. Since the type of breed we had given, it produces the brown shelled eggs. And since they are the free range eggs, they get a premium price for it. So the farmers told us that we sell these eggs at the rate of 7 to 12 rupees per egg. But again, for calculation purposes, we take only the market rate. The table egg in our place is for rupees 5 per egg. So by taking this rate, rupees 5 per egg, so they have been able to generate 16,20,000 from the sale of eggs. And then spent hens, once the hen completes that laying cycle, it is called as spent hen. So this usually the rate for spent hen is 150 to 200 rupees per hen, but for calculation we have taken only rupees 100 per hen. So it comes to again 2,16,000 rupees. The total value which got pumped into the economy of the area was 18,36,000. So investment on the whole project was 8 lakhs, and the economy generated was 18 lakhs. So that is what the PESA more than double in just 2 years, what we are trying to convey. So these are the tangible benefits, those benefits which we could rather calculate in terms of money. Then there are a lot of intangible benefits, those that cannot be converted into money. That is, suppose availability of eggs at home. When the eggs are available in the rural area, that strengthens the nutritional security. The women get empowered because they get money through sale of eggs, with which they fulfill their daily needs, and this enhances social and economic security. Then they scavenge these hens, they scavenge on the kitchen waste, worms, insects, fallen grains, and debris. This creates the cleaner surroundings, and they are able to utilize those available resources. Then women folk and aged people, they look after these birds. Thus, we are able to utilize otherwise unused or underemployed workforce. There is increased local production of eggs. This decreases dependence on imports and it strengthens the local economy. And there is an increase in soil fertility because hens scavenge on weeds and insect pests, and their droppings they add as manure to soil. Then this was about the natural farming, which we did in a different way. Then the second intervention, this is about the nursery raising. You see, wherever we carry these poultry development programs, what we do, we try to augment the supply of germplasm. We give a good quality germplasm to people so that they can rear those good quality birds at their home. So for this, there are nationwide, there are various programs under ICR's All India Coordinated Research Project on Poultry Breeding. They have developed various breeds for these backyard poultry farming, like Van Raja, Gram Priya, Serenity, and so on. There is a long list, and newer breeds are being developed. Then there is ICR Poultry Seed Project. In various states, this supplies those good quality birds. This ICR Poultry Seed Project has a mandate of distributing this germplasm, and then there are state poultry development programs. They run various rural backyard poultry development programs under which they distribute the chicks. Now, this dissemination of improved germplasm can be done in three ways. One is we distribute the hatching eggs among the farmers. This is the cheapest intervention. This needs very less money. Or we can distribute day-old chicks among the farmers. Or third option is this: we distribute 3 to 4 week old chicks among the farmers. Then, if we distribute hatching eggs, this is the cheapest intervention. But problem is that when we give them hatching eggs, then they have to search hen, which will hatch those hatching eggs for them. Because nowadays those improved breeds of hens, they do not go broody. उनमें वो क्षमता नहीं है कि वो अपने अंडों पर बैठ के उनमें से चूजे निकाल सके. That thing, that trait, that characteristic of hens is now lost. Nowadays, new breeds of hens do not have that broodiness character. If we distribute day-old chicks, again we have a problem. It is a little, although as compared to hatching eggs, it is a costly intervention, but still it is a cheaper intervention. But again, who will take care of those day-old chicks, because hen is not there? Then the vaccination of those chicks is difficult. We cannot, because those chicks will be spread here and there in a village, and one has to go from one home to another home for vaccination. This 3 to 4 week old chick, this is, we call them as nursery raised chicks. In this system, we rear those chicks for 3 to 4 weeks at a poultry farm and then distribute those birds. During this 3 to 4 weeks, when they are in the farm, in thousands they are there, they may be a government farm or a university farm. There, the vaccination, etc., is carried out, plus they get those necessary heat वगैरह उनको चाहिए for that initial 3 to 4 weeks. They get that, so there is a little risk to the farmers. Though this is a here the success is more, the farmers do not face much problem, but the problem comes to the university or the government farms where there is mostly limited space available. There they cannot rear too many of these chicks at their farms. So here, what we have done, we have tried to outsource this brooding of chicks to some farmers. We have made those farmers as a sort of a nursery farmers. We give them the day-old chicks. They even initially we gave them, then they later on they purchase those day-old chicks. They rear it for some time at their farms, and then they distribute it. They sell it to the farmers who are desirous of keeping those chicks. So when we tried to work out the economics of this, some of the farmers who indulged in this nursery raising of the chicks, we found that their earnings per bird was some rupees 24 or 25 rupees earnings, and that too in 4 weeks they were able to sell those. The cycle here is in broiler farming it is a 6 weeks cycle, but here it is 4 week cycle only. So they get good earnings by selling the those when they rear it for 3 to 4 weeks, then sell it to the farmers. They get the earning of rupees 24 to 25 rupees. Is a good handsome earning. So many of the farmers, there is a list of various farmers who have taken chicks and then reared them for some amount of time, and this success story continues. So this is another second. The third one we will be discussing is this horticulture, poultry horticulture integrated farming system. This we have tried. In Kashmir, we have a lot of horticulture. Apples of Kashmir are famous. You have very, very big apple orchards. So apples grow on the trees, and then the floor of those orchards, that is, there is some grass grows there and everything. So we try to integrate these poultry rearing in those apple orchards. We have some 1.6 lakh hectares in Ghent under orchards, area between these trees that remains underutilized. So when we allow the hens to scavenge in those fruit orchards, this decreases the feeding cost. So hens will feed from that whatever the insects, whatever the grass, and whatever other things they find from that orchard. They will eat those things, so there will be no cost on feed. This will boost the local poultry production. This will optimize resource utilization and thereby increase the farm income. There will be other advantages like manuring, dividing of those orchards, and these again hens will play some role in disease management by of the fruits, because they will eat those pests that will cause otherwise disease in the fruits also. So in that way it will be beneficial to the fruits also. So here we have tried to calculate the cost benefit ratio, and we have tried to compare the benefits from a there is a same orchard of same size. Then it is again they go for on the orchard, fruit orchard, and on the other side we have integrated high density orchard on the same plot of land. So when we tried to find the benefit cost ratio, it was 4:1. Then it is alone the orchard of fruit orchard only, and when it is integrated with chicken, it is 4.4:1. So we have an additional income, and there is an increase in the farm income in from the poultry. Then we have tried one more innovation. As I told you earlier, that those new poultry varieties, they have been selected against rules. They do not, they have lost that capacity to sit on the eggs and produce chicks, what earlier we used to. So the farmer always depends nowadays for some agency to supply the chicks. So if he would like to produce eggs and then hatch them at his own home, for him we have done some innovation. Nowadays we have a modern hatcheries. They are very big hatcheries. Their capacity is from thousands to lakhs, and it is very technologically very intensive, and you need a lot of money on investing on those very costly machines. There are mini incubators also available. These mini incubators, they are imported ones, and they are also quite costly. Their capacity is 10 to 50 eggs. A farmer could keep them, but they are very technologically very intensive, and they are, in terms of capital or in terms of cost, there is a very high cost. What we have done, we have fabricated our own, we call it a Shalimar Easy Hatch. It is we have fabricated our own incubator, which is portable, which is cheap, which is fully automatic. It has a very limited, small size dimensions of 36 centimeters into 28 centimeters to 15 centimeters. It weighs just 1.3 kg. It can carry 30 eggs. It maintains required temperature and humidity. It has even automatic turnings also. And power consumption, you can see here, is just 60 watt. So you can run it on the inverter if you have. So farmer can easily use it. And you know the cost. One of the very good thing about this is cost is just 1800 to 1900 rupees. It is by far it is the cheapest fully automatic egg incubator available, and with hatchability is more than 70%. We have recorded in this. This is the one more intervention. And then third, we were talking about the diversification of the poultry. As Dr. Mishra was also saying in his talk about, he was talking a lot about ducks. I will just draw your attention towards another waterfowl, that is geese. It is a sort of a bigger than duck, and we find it in good numbers in Kashmir. So we have in our we have tried to characterize it. We have tried to study it, uh, its various production performance, its variations, whatever are available, and we have been able to register this with uh, National Bureau of Animal Genetic Resources. This is in fact India's first and the only geese breed that has been so far registered. First and the only one in India this has been registered. And then one more intervention. Uh, Dr. Mishra talked about this sexing. He was showing you how we identify the male chick and the female chick through vent sexing and other things. In geese also, they look so alike. The male and female is from their morphology. They look so alike that you cannot identify which one is male and which one is female. We have tried a very novel way or a practical method of gender identification in genes, that is by recording their sound and then subjecting it to the sound analysis. We generate an audiograph. Audiograph is nothing but when we put we use this program, computer program. It is again a freeware. It is freely available. It is not a paid program. It is freely available program on the internet. So when we put that sound, recorded sound in this software, you get an audiograph, and for this female geese, you will get this type of audiograph. For the male geese, you will get this type of an audiograph. So by just seeing the audiograph, you can just identify which bird is male and which bird is female. So this a novel approach we have tried to generate to separate out. This is very, very important. Why it is so important? Because farmers, these geese, otherwise we rear them, it is very, very costly to rear the birds, and farmers are interested that they want to sell away the males. Then for the whole of the year, these, they are feeding themselves in those lakes. We have a lot of lakes here. But when the winter comes, they come back, and the farmer has to feed them at his home. So he cannot afford to keep all the males. At that time, he disposes of maximum males. He keeps few of the males and most of the females for the breeding purpose, for the producing the next crop. But to identify those males and sell them, that is very difficult task. Only the old people who have, by their experience, probably their ears are trained and they are listening to the sound, and then they are saying that this one is the male and this one is the female. But here we have given them a practical method of gender identification in genes. Then coming to this cold conditioning in poultry. Why it is so important? Because I told you that we are living in a cold place in Kashmir. There we in the winters particularly, we have to use a lot of fuel. we have to use a lot of energy so as to maintain the temperature in the poultry house, and that only then the poultry will grow well when the temperature is maintained. Otherwise, these chicks will suffer because of the cold. There will be stress on those. They will not grow well, and then there will be lot of problems. What we are trying to do through cold conditioning is that we are trying to accustom them. We are trying to make them used to this cold. Not this we are this accustoming or this cold conditioning. We are doing both in the pre-incubation period as well as in the post-incubation period. See, we have tried what we are doing in pre-incubation period. Those chicks, those eggs which we are supposed to hatch in the hatchery, we are exposing them to the cold stress during the incubation period. We tried various cold stress period exposures, like on day third, or sometimes we tried repeat exposures on third, fourth, and fifth day. Then these were the post-hatch cold stresses. Then pre-hatch cold stresses on the 18th day of incubation. We tried various exposures, and then finally we were able to find that the post-hatch cold conditioning done on third day of life as a single exposure, this augments cold conditioning in terms of improved FCR, better economics, and decreased mortality. This cold tolerance, actually, where from it comes? This acts as a acts on an epigenetic level. There is down regulation of certain genes which happens because of this exposure to the cold. So we have tried to quantify this HSP gene expression, like the heat shock protein 70 gene. This gets expression when the birds are under stress. So here we have tried to quantify that, and we have found under certain pre or post hatch cold conditioning exposures or treatments, these they have got down regulated, and in certain conditions they have got up regulated. And this we have then found that actually the post hatch cold conditioning done on third day is the most. The other exposures were also beneficial, but the most beneficial was on the third day of life, a single exposure during the post hatch. That those birds are able to have better FCR, better economics, and lesser mortality in their life. So, summing up all those things which we have discussed so far, is that the bioeconomy is our need for future, as well as we will exhaust our non-renewable fossil fuel resources one day, and we have to sustain our growing global population simultaneously. The enhancement of productivity of agriculture system using sustainable approach will be required to meet growing demand from livestock and farming systems without drastically increasing their environmental footprint. The interventions which we have discussed here, like backyard poultry model, nursery raising, hortipoultry model, low-cost incubator, or diversification of farming system, or cold conditioning, they have a very important role in reducing the environmental footprint and ensuring sustainability along with productivity and profitability. Thank you very much. I end my talk with this goal that sustainability is the future economy. Thank you.
Thank you so much, sir. Thank you so much for your kind words and sharing precious knowledge with us. Definitely, this will be beneficial for all of us in future aspects. Now, moving ahead, it is time for paper presentation. And I would like to invite our technical session chair, Dr. Anubha Pandya ma'am, Head of the Department of Chemical Science, Christian Eminent College, Indore. Ma'am, shall we start our presentation?
Yes, ma'am.
Anubha, okay. With the permission of session chair, I would like to invite our first paper presenter, Ms. Yanshi Nigam, Department of Bioscience, Christian Eminent College, Indore. Ms. Yanshi, is my voice audible to you?
I think
she is not Yes,
Yeah, right
I
now
have
she
checked.
is not.
Not. I think some technical error is there. So now I would like to call our second paper presenter, Ms. Palak Joshi and Shubhanshi Sisodia, BSc student, Department of Bioscience, Christian Eminent College, Indore.
Yes, ma'am. Second.
Okay.
Ma'am, is my screen is visible?
Screen is visible, but your presentation is not there.
Oh, okay, ma'am.
Yes, it is visible.
Okay, ma'am. Is it visible,
You
ma'am?
can start. Yes, yes, you can start.
Okay, ma'am. Good afternoon, all of you. Myself, Palak Joshi, and myself, Shubhanshi Sisodia. We are representing our college, Christian Eminent. Our topic is recent approaches of biological sciences for enhancing the pharmaceutical researches for human welfare. As we all know, biotechnology means the utilization of biological and engineering principles to enhance researches and productivity by using microorganisms and their derivatives or modifying products for human welfare. Advancements of biotechnology tools have been widely employed for the laboratory researches, pharmaceutical, healthcare, agriculture, food industries, etc. Pharmaceutical companies are widely utilizing biotechnological tools and techniques for the manufacturing of drugs, pharmacogenomics agents, genetic testing, and disease diagnosis. Here are some parts of biotechnology. These are cell structure, molecular biology, monoclonal antibodies. Molecular biology includes DNA technology, genetic engineering, and tracers. In DNA technology, banks of RNA and DNA and proteins are included. Complete map of the human genomes are also included. In genetic engineering, synthesis of new proteins, new antibiotics, new types of plants and animals, gene cloning, mass production of human proteins, resources bank for rare human chemicals, gene therapy, etc. are included. In monoclonal antibodies, diagnosis and anti-cancer drugs are included. Cell structure, transfers of new genes into animal organisms, culture of plants form, single cells, etc. are exist. Biotech companies are making biotechnological products at molecular level by recombinant DNA technology, which entails genetic manipulation of cell through genetic engineering principles. The hybridoma technology is a technique used for the production of monoclonal antibody as a magic bullet in the treatment of several diseases. Biotech companies have designing and producing safer vaccines by various manipulated organisms which are transformed through genetic engineering by minimizing the risk of infections. This table is shown the list of pharmaceutical proteins, that is hemoglobin, and diseases anemia, asthma, and the recombinant protein is interleukin receptor. Blood clots, and the recombinant protein is tissue plasminogen activator. These products of many pharmaceutical companies are widely using for the prevention, diagnosis, or treatment of many types of diseases have been made through biological aspects. Pharmaceutical companies are widely utilizing biotechnological tools and techniques offer the manufacturing of drugs, pharmacogenomics agents, genetic testing, and disease diagnosis, etc. Study of genetic variation underlying different responses of drugs. These are medical applications, approved drug products. Activase is for heart attack, diabetes, by insulin, etc. The more important benefit of pharmaceutical biotechnology leads in the form of better vaccinal immunization. Now I'm over to you, Shubhanshi. Shubhanshi, please continue.
Now I am talking about vaccines. Vaccines, a substance used to stimulate the production of antibodies and provide immunity against one or several diseases. Um, Prepared from causative agent of a disease, its product or a synthetic substitute treated to act as an antigen without increasing the diseases. Vaccine. These are inactive, attenuated, purified, lived, or dead pathogenic metabolites, toxins, toxicoids, proteins, polysaccharides, which can generates immune responses for prolonged immunity as vaccinal immunization. These are mainly three types: dead bacteria or inactivated virus, live on virulent or attenuated strains, viral fragments of bacteria molecule as subunit of vaccines. These vaccines, also known as conventional vaccines, are widely accepted for many diseases: smallpox, chickenpox, cholera, typhoid, tuberculosis. Recombinant vaccines. Recombinant vaccine is a vaccine produced through recombinant DNA technology, such as bacterial surface protein. Recombinant vaccination is a preventive mode of immunization through modern generation of vaccines with the addition of manipulated genes to produce specific proteins or metabolites as vaccines. There are several modern strategies to establish a novel recombinant vaccines. There are of two types: subunit recombinant vaccines. These are components of pathogenic organisms, including proteins, peptides, and DNA particles. Second, attenuated recombinant vaccines. These are genetically modified organisms like bacteria and virus, which are modified in such a way that they could only produce immune response but not disease. Recombinant vaccine, which developed to human healthcare. First, acute respiratory disease. It is a viral disease and caused by influenza A and B. The second one is AIDS. Why it is also a viral disease and caused by HIV virus. Um, Liver damage. It is a viral disease caused by varicella. Tetanus. It is a bacterial disease caused by Clostridium tetani. And pneumonia. Pneumonia is a bacterial disease. It is caused by Streptococcus pneumonia. Thank you.
Thank you so much, Palak and Shubhanshi. It is requested to Dr. Anubha, ma'am. Ma'am, any question from participant?
No, ma'am. No.
Thank you, ma'am. Thank you, participants. We have some time limit, and due to this, we will continue another presentation in tomorrow in our second technical session. So, moving ahead, now it is time to express our gratitude towards our guest speaker, session chair, and all participants. And for that, I would like to invite Professor Ekta Rawat, Madam, Assistant Professor, Department of Bioscience, Christian Eminent College, Indore, for vote of thanks. Ekta, ma'am.
Thank you, ma'am. Very good afternoon to everyone. Respected dignitaries, respected principals, sir, invitees, participant, and their student. It gave me an immense pleasure to deliver the vote of thanks on behalf of Christian Eminent College and organizing committee. I, Ekta Rawat from Department of Biosciences, I am very hearty gratitude to our today's keynote speaker, Dr. Azmat Alam Khan, Professor cum Chief Scientist, for sharing their valuable views with us in first day of international conference on topic novel circular bioeconomics model for sustainable poultry production. Thank you so much, sir, for such a wonderful and informative session. I sincere thanks to Dr. Saurabh Jain, sir, Chairman and CEO of Research Foundation of India, for their collaboration and support. I also sincere thanks to Honorable Director Achin Gupta, sir, Honorable Admin Director Y.S. Chauhan, sir, Honorable Principal Dr. Rajesh Vyas, sir, Honorable Vice Principal Rajesh Shah, sir, to motivate us and even guide us to organize such an international conference. I want to thank head and faculty of various departments for their contribution. I would like to thank C.K. Tiwari sir and Amit Nilose sir for their technical support. I thank all the participants for your presence and patience. Once again, I thank one and all present here. Thank you so much. Have a nice day. Thank you.
Thank you, ma'am.
Hello. Yes, ma'am. Please continue. Please continue. Please continue.
Yes, sir. Sir, please, can we leave?
Yeah, it is the time to wind up this session. And in the same day, same platform, we will meet tomorrow at 11 a.m. So I welcome you all once again. Thank you very much. And now it is the time to leave the session. Please, thank you very much to all of you.
Thank you so much, sir. Thank you so much.
Thank you, ma'am.
Congratulations, sir. Congratulations, team.